That's the lavalier. Test, 1, 2, check, 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 check. Mic check, 1, 2. Mic check, 1, 2, 1, 2, check, 1, 2. I am checking this microphone. Check, 1, 2. A, 1, 2, A, B, C, D, E, F, G. Peter Piper picked a peck of pickled pecklers of fish.
It's peaking extremely.
Many. Here, we can turn it down.
But that's not it. Like, your voice isn't loud at all. It's
Check, check.
The signal is peaking.
Check. Mic check, 1, 2.
The level looks okay. It doesn't
Check.
need phantoms.
Mic check. It's just a noisy environment, maybe.
Okay, maybe if everybody shuts up, it works.
Here, I'll just...
Can you, like, the audience would have to hear us. Does that work? Like, with the speaker. Are we piped in through the back?
Well, it's the same output.
Yeah, no, I'm just saying, like, the volume for the room is on. Can anybody even hear what you say through that?
I don't know.
That's what we're trying to get to do.
Well, here, let's get this one working. 1, 2, mic check. Check 1, check 2, check 1, 2. Mic check, 1, 2, 1, 2. Check, 1, 2. A, B, C, D, E, F, G. 1, 2, 3. Maybe. This has nothing to do with that.
No, it's my output. I'm not feeding the amp directly, right? This is going by the mixing console for the house speakers.
Yes.
Yes, I'm just like, I can't hear anything you're saying in the room.
I understand. Okay, so you have to turn on monitoring on that particular port. Check 1.
No, like, I can hear you.
Yes. No, I understand that. You need to set up monitoring to go out through there. That's what it is.
That's already done.
Check, check. Mic check. 1, 2. You're on. I'm still on. Oh, there we go. Check. No. Which mic is that?
That's this one.
It's not working. Check. Oh, there we go. Ooh. That doesn't sound good at all. No, that's... Check. Mic check. 1, 2. That's better. Yeah. Check. 1, 2. Mic. Mic check. 1, 2. Check. Hello. Checking 1, 2. A, B, C, D, E, F, G. A, B, C, D, E, F, G. 1, 2, 3. A, B, C. So very peaky. Why is that so peaky? Click this here. Hello. This is how we will be using these microphones. So if I'm speaking normally, then that kind of works.
should work.
It'll be good enough. It's very loud in here.
Yeah.
Okay. And if I take this off, or I'll mute this. Check 2, check 1, 2. That one's much better. Ooh, that's still got some, no, that's muted, so it's got to be this. Do you have another microphone on? Are you sure? Check 1, 2. Maybe if I'm over here, is it better? Maybe it doesn't squelch as much here. Okay. So it's just being over there in front of the mic, in front of the speakers. Okay. That'll have to work.
Where's the control? I've got my hand. And my mic is working? Lights are good? Good morning, everybody, and welcome to the 30th annual BC Marine Mammal Symposium. It's been actually going back over just 2 years since we were last here. We've got a huge number of people online. We purposely, because we're not 100% back to normal, we're about 80%, and being very aware still that we are still in a pandemic, so we did restrict the numbers this year. And so those that are in the room feel very fortunate you got some of the few seats that we allowed to have in person this year. We know many of you were disappointed and are watching us online, plus we have people from literally all over the world that are joining us right now. We've had a few challenges getting us plugged in and sorting out the new system because during COVID, UBC also rewired some of our AV equipment here. So we had a few surprises despite being here at 7:30 this morning to get everything ready. A few things to go. We've got a really long program, which is a sign of just how much research is happening here in British Columbia on marine mammals. This was an event which we set up 30 years ago to bring people together to share their research findings in a semi-informal setting. It's not your traditional science conference. In other words, you don't get a full 15 to 30 minutes to talk about all your methods and everything else. The idea here is to reconnect with each other, to share information in literally 5 minutes, and then to have time during the breaks or outside of this meeting to connect, to ask all those fine details that you want to know about or you want to share with others. A few pointers for speakers. When you come up here, you're going to have this really blinding light here. You just have to block it out. And that is so that we can talk to people in line and show up properly. We are being mic'd. The speakers will have a lapel mic. Rob will be instrumenting you up with that. Kate has it on right now, ready to go as our first speaker. We have presenter view. You'll see slides on here. It may look quite different than what you're expecting. There is a control. And again, for those speaking after the break, you're welcome to come up and check it all out. Otherwise, just trust me and trust our team here that it is going to work. We have one thing in past years. Yes, we were just pointing out our key support people. In past years, sometimes we've had people that have trouble dealing with the 5-minute rule. And yes, we have 5 minutes plus 2 minutes for questions, but sometimes even I've learned standing up next to people to encourage them to turn it off doesn't work. So we used to have our rooster. Unfortunately, during COVID, our rooster lost its voice. So now we have the big hand. And so until we can resurrect our rooster, so please try to stick to time. We have a lot to get through. We are starting late right now, but we do have a fairly flexible schedule as well. I think that's all we've got right now. I'm going to ask Kate to come up. And as our first speaker, she's going to be talking to us about biologging data and the Pacific Coast Feeding Group of gray whales.
Okay. So good morning, everyone. Today I'm going to be talking about using biologging data to estimate the energetic cost of foraging in Pacific Coast Feeding Group gray whales. And so a little introduction to gray whale ecology. These are benthic feeding baleen whales, and you can tell that by the sediment plume shown in this figure or in this photograph that these whales are indeed foraging. Depending on which foraging ground they're using, they feed on benthic crustaceans, either amphipods or mysid shrimp as the dominant prey species. There are multiple stocks of gray whales shown on this map. We're going to focus on the dark blue side, so the eastern North Pacific. This population is about 20,000 individuals. They breed in the lagoons of Baja, Mexico, and migrate up to the Arctic to feed. And then there's also the western North Pacific population in the light blue. You'll notice that the Pacific Coast Feeding Group gray whales are not shown on this map, and that is because their situation's a little bit more complicated. You'll notice that the purple box showing their summer foraging range in the waters of the Pacific Northwest includes both US and Canadian waters. In Canada, this group is recommended to be recognized as endangered, whereas in the States, it is seen as a seasonal subset of the eastern North Pacific population. The PCFG and the eastern North Pacific both breed down in Mexico. And so what my project is looking at is the foraging energetics. And so we know about energy requirements for the eastern North Pacific and western North Pacific population, but these models don't include this PCFG group, which we've seen has a different migration strategy and different prey than these Arctic foraging whales. And so we really need population-specific parameters for this Pacific Coast feeding group in order to understand their energy needs. And so to do this, my project is using data from suction cup biologging tags, of which we have 12 tags deployed that have about 92 hours of data collected so far. And these suction cup tags are kind of like if we put a Fitbit on a whale. So they've got the 3D sensors and can track movement, and you can use that to estimate energy expenditure. So what this data looks like is something like this. This is a track coming from one of those biologging tags. The blue section represents when the whale is at the surface, whereas the yellow is when the whale is down at the bottom of the dives. The top panel is showing the dive profile for the deployment, with the blue box showing the section of the deployment that is seen in the track on the slide. And so if we animate this, we can see the whale moving. So the whale's coming up to the surface now and going down to the bottom. Notice that the whale keeps going down to the same spot, which indicates that this whale has likely found a pretty solid prey patch and is foraging. And so with this, we want to know, can we separate out these different behaviors in the tag data, and how much energy is the whale expending in these different behaviors? And so that brings us to the research questions for my project, which is looking at the energetic cost of foraging at 2 scales. One at the dive scale, which is looking at the behavior states, so if the whale is searching versus foraging versus transiting, and one is in the roll events, which are these specific foraging tactics that make up the foraging behaviors of this population. And so to start with the foraging tactics, what you're seeing now is a video from one of the tag deployments, and I want you to pay attention to that red line in the bottom panel. The whale is about to go into a roll event, meaning that it's turning onto its side, and you'll start to see a bunch of sediment coming up into frame in the camera, which leads me to believe that this is a foraging tactic for this whale, and that if I look at all of these roll events, that is going to encapsulate all of the foraging tactics of this population. And so when we get those roll events classified based on their body position, so the axis that's in the horizontal plane of the page is looking at the median roll, so how much the whale is on its side. The axis going back into the page is looking at the pitch of the whale, so if the whale's nose is more towards the sediment or more towards the surface. And then that vertical axis is looking at the duration of that roll event. And so when we look at the data, we can see pretty good clustering there, which is indicative of, one, we can actually find these different foraging behaviors in the tag data, which is super exciting. And two, that if you notice, this red cluster seems to have longer durations than the other two. And that can have an energetic implication here, with the being able to perform the behavior for longer, meaning that it is less energetically expensive. Moving on to the behavior state aspect, so at the dive scale now, I'm going to be using hidden Markov models to try to classify these different behavior states. And while this work is still in the preliminary stage, we can see some visual patterns in the tag data. So this first panel is looking at the depths of presumed foraging versus presumed transiting dives, and you can see that the foraging dives tend to be deeper and longer than the transiting dives. If we look at roll, so again, how much the whale is moving onto its side, you can see that the foraging dives have higher roll than these transiting dives. If we look at pitch, which is again the whale's nose orientation towards the sediment versus the surface, there's much more variation in the pitch during the foraging dives than the transiting dives. And this last panel is what's really exciting and how I'm going to be able to get at the energy expenditure. Each of those lines is in a different color, and that represents one of the 3D axes of this tag data. And you'll notice that there's a lot more variation in that signal for the foraging dives versus the transiting dives, which tends to suggest that these foraging dives are going to be more energetically expensive than the transiting dives. And so just a reminder of why this work is important, you're seeing a tag video here where a boat narrowly escapes hitting the whale, and so the whale is swimming through its wake right now. And so this just kind of brings into very stark reality that we need to be able to quantify the impact of these threats and these disturbances on this population, and this biologging data is a great way to do that. I'd like to take the time now to thank my committee members and the GemLab Granite Field team for all their hard work collecting the data. Also a big thank you to John Kalambakitis for data sharing, assisting with tagging logistics, and allowing us to operate under the permit. So thank you all for listening, and enjoy the rest of the day.
Anybody with a question? If not, Mariana?
Were you able to also collect your heart rate? Because you were saying it's kind of like a Fitbit, but it's not. Can you collect heart rate from your?
I'm not sure that's mic'd or not.
It is? Okay. So the question was, can these tags detect heart rate? And not the tags that I've been looking at so far. We did have one tag that had a heart rate monitor in it, and so that is going to be really exciting to look at. I just don't have that data processed yet.
Exciting. Thank you.
Thanks very much, Kate. I think we're ready for Stephen Rafferty.
Good morning, everybody. It's really a tremendous pleasure and privilege to have the opportunity to participate in this meeting, and the focus on the discussion is actually going to be a brief overview of some of the pathology that we've been trying to identify in gray whales that have stranded along the west coast. And Kate gave a tremendous introduction and overview of the natural history of these animals. They tend to, in the summers, spend their time in the north in very vibrant feeding areas, and then will migrate south for the winter to calving beds. And the image on the far right will actually show you some of the population trends, and these have been tracked since the 1960s. And what was really kind of interesting is that since 2016, you might appreciate that there was a peak in the population of about 28,000. And just within the last 6 weeks, there was a publication out of Southwest Fisheries Science Center by David Weller and others that the population over the last 6 years has declined to about 16,000. So there's been a 40% loss in the number of these animals over time. And this is not unprecedented from a historical perspective. In fact, in 1999 and 2000, you'll see that there was also another loss of about 30% of the population in that time frame. And then there are anecdotal reports of mortalities in the mid-1980s as well. Those animals that died in 1999 and 2000, there were about 3 that were actually necropsied, or what we call necropsia, post-mortem examination, a couple in California, and then we wound up doing one at IOS on Vancouver Island. So this time around, there was actually an intent to try and improve the response and the data that would be gathered from this particular event itself. You can see that the decline actually started in 2016, but it was the official declaration of an unusual mortality event, and this is something that's made by a working group with the United States, NOAA, National Marine Fisheries Service, and essentially it allows funding in order to coordinate a response. And in this particular case, we're actually talking about transboundary related incidents. So there are collaborators from Mexico, Alaska, and we were even in contact with investigators from Russia looking at the eastern population of gray whales to see whether there were similar changes that might have been going on there as well. See, the peak mortality occurred in 2018, 2019, and then the demographics or the breakdown by country. In Canada, although we only had 25 animals, we must have posted about 22 or 23 of these individuals. In Mexico, there were 280 animals, and there were only 4 necropsies done, but 2 of which we would classify as being full examinations. And certainly in the United States, there's a much more intensive effort and more resources available and expertise in order to be able to undertake these examinations. In fact, there were a total of 606 individuals that were lost, or that were identified, sorry. So of those that died, there were 606 where they were identified by either member of the public or a regional stranding response program, research scientists and biologists. And in the United States, these are documented as a level A information. So essentially it's the tombstone information, the gender, the age, size. Oftentimes these may be accompanied by photographs that allow some interpretation of at least some external features of the animal. The map on the right actually shows those animals, the distribution of those animals where these level A data were reported as well. Oops. It doesn't seem to be advancing. And so there were a couple of hypotheses that were generated. And first of all, the unusual mortality event itself actually extended from Mexico right up through to Alaska. It was designated in 2019, but just keep in the back of your mind that the initial mortalities were incrementally increasing to a peak in 2019, but they originally started in 2016. Some of the clinical signs, and these were very apparent, particularly in the calving grounds in the Mexico lagoons, where there were juvenile animals or non-pregnant females that presented in very poor body or suboptimal body condition. There were actually fewer calves that were observed as well. In some areas, they would typically have over 120 calves in a particular lagoon, and in fact, they'd have fewer than 18 or only 20 individuals, and that seems to be persisting even through to this time. The pathology findings of those animals that have been examined, they tend to fall into 3 or 4 major categories. There's malnutrition, and I'll give you some examples of this a little bit later on. Human interaction, which we would define as vessel strike predominantly, so vessel or propeller strike. Killer whale predation, and this is something that really wasn't on our radar at the initial stages of the investigation. The killer whales are certainly recognized in Alaska. There are specific ponds that seem to target some of the calves from gray whales and can take up to about 50% of the calf crop in a particular area. We've also, specifically in British Columbia, have been screening for different disease agents that we would associate with either reduced fertility, increased mortality, or even degrees of morbidity. And so viral pathogens would be the morbillivirus, brucella. We were even looking for influenza and a couple of other ones. As well, we've had a long-term survey looking at what we call harmful algotoxins. These are the domoic acids and saxitoxins. They've long been recognized within cetacean species, but their impact hasn't really been fully resolved. They may very well just transit the gut. They're ingested, they transit the gut, but may not necessarily be assimilated and exert any sort of pathogenic effect. Our preliminary consideration with regards to the loss of these animals was that in fact it was very likely multifactorial, just because of the disparity or the number of different findings that were identified with the postmortem findings as well as field observations. Environmental drivers that may very well be leading to malnutrition, and I'll give you some examples of this a little bit later on. Killer whale interactions or human interactions, and then the killer whale predation. This first example is actually the malnutrition, and increasingly we were finding a number of these animals. We went through and actually scored all 60 individuals with signed case definitions, so we were able to rank them from 1, 2, 3, and 4, 1 being an example of the animal on the far left, or your far right there, where essentially you can see bilateral concavity or indentations of the musculature along the entire length of the animal. Often this is associated with infestations of some of these ectoparasites. Essentially these animals are swimming a lot less slower. They probably have a degree of immune incompetence and debilitation, so they're essentially being colonized by these organisms. The map itself is actually trying to, or it shows that those animals that presented with most severe emaciation were actually in Washington State. So essentially the hypothesis is that they're feeding up in the Bering Strait. They were able to make it down as far as Mexico, and they were actually dying, presenting in this malnourished condition, predominantly in Washington State. We do have a couple of individuals that were identified in British Columbia as well. At one time, it was thought that gray whales only fed in the Arctic, and then they were essentially not feeding as they migrated through down to Mexico. But in fact, that's been disproven, and there are feeding grounds where they do aggregate throughout the coastal corridor itself. But what was really striking was from an environmental perspective. You look at the map, and the image on the right was, on the left, sorry, was from 2013, and you could see the extent of the ice coverage throughout the Bering Strait and into the adjoining waters, whereas in 2016, they actually noted a very advanced loss of ice. This was the lowest ice cover that had been recorded in history. There was a loss of about 50% of the surface area, about 75% of the volume. This is advantageous to the animals in that they have access to greater feeding areas, but it's a disadvantage in that the consequence of low ice cover is that there is suboptimal prey that's available. The graph on your far right is from a paper published by Sue Moore, a biological oceanographer with the University of Washington this past March. And I don't know if I can show you, but the graph on the top left showing the decline actually shows that there's not only a decline in the prey abundance, but the quality of the prey itself is also depleted. So our working hypothesis is that these animals are just not attaining a sufficient prey. They're not attaining a sufficient level of nutrition in order to conceive a calf and then also to sustain pregnancy through to the migration north. The next image, and this is the examples of the killer whale predation, and just showing that it tends to be congregated around Alaska, off the BC coast, and down in California. There are actual defined case definitions now, as well as scores, whether it's probable, suspect, or confirmed predation, depending on whether the nature of the lesions. This last incidence is that these animals follow the same corridor as many of the shipping lanes, and increasing we're finding numbers of animals that present with catastrophic injuries. These are not invariably lethal. You can see the image where there's serrations along the surface of the skin. Although it may not cause immediate death, there's a portal for secondary bacterial involvement. And so just in conclusion, we're still looking at trying to determine what are the primary drivers, whether this is ecosystem-based changes or related to carrying capacity, the prey availability, some of the contributing factors, and obviously killer whale predation was something that was noted before 2016. Whether it's increased over time is difficult to infer, but that's an active area of investigation at the moment. Similarly with human interactions. And we actually have been working collaboratively with a group in UC San Francisco looking at microbiomes and trying to determine whether there are in fact infectious agents associated with the strandings. And lastly, I just need to acknowledge all of the collaborators, and obviously this is a multidisciplinary, multi-institutional team that we're involved with, and a lot of different experts that are contributing some really valuable information. So thank you.
Stephen, if you want to stand over here. Do we have any questions? Go ahead, we've got two. I'm going to hand you this mic.
In regard to, in the beginning there, you mentioned that there was this, not quite a quasi-cyclical sort of population trend in gray whales, but something that has shown up in the past as well, and they've recovered. What do you project would be the effect of long-term climatic changes on the planet on this trend? Are we expecting them to continue to decline, or are we looking at a potential possibility of recovery again?
The question is related to climate change and the potential long-term impacts, and whether it's something that's going to progressively deteriorate or whether these animals will be able to recover. And the simple answer is I don't know for sure. Historically, the changes in climate would have been very gradual, but what they're talking about now are stepwise changes. So these are abrupt, they're transformative, so there are impacts as a consequence, and whether these whales will recover in this particular situation, I don't know, but they certainly seem resilient based on past observations.
Tom, a quick question?
Yeah, could the instances of increased killer whale predation also be related to malnutrition in some way? Like, for instance, weakened whales would be easier prey for killer whales?
Absolutely. Absolutely. The question was whether some of these animals that present in poor body condition may predispose them to killer whale predation, and that's absolutely a scenario that's being looked at. Unfortunately, we don't have enough data to do the actual statistical analysis on that, but it is something that's been considered.
Thanks, Rob.
Anyway, thanks very much, Stephen. And our next speaker is Beth Volpov.
Okay, thank you, Andrew. Hi, my name is Beth Volpov. I am a postdoc here at the Marine Mammal Research Unit at UBC. And today I'm going to be talking to you about some work we're doing where we're using drones to look at the movement, behavior, and physiology of resident killer whales. I like to start my talk off by doing something that's normally reserved to the end and thank my collaborators and everyone that helped collect this data. This data was collected in 2020 by the fantastic crew you see here, many of which I also see you guys in the audience as well. So thank you very much. So the overall problem that we're investigating is we want to know the energetic expenditure of these killer whales, because ultimately all physiological activities require balancing supply with energetic demand. But we can't measure metabolic rate directly in the field. And we need accurate estimates of metabolic rate to predict food requirements of these animals and energetic models. One tool that we can use to indirectly estimate metabolic rate is an activity budget. An activity budget is simply the proportion of time an animal spends doing different activities or behavioral states. And this is important because different behaviors require different amounts of energy. You are using less energy now as we are sitting here inside the symposium compared to if you were to go run a marathon or something like that. So that's why there's a focus on activity-specific metabolic rate instead of just an average over all the activities. The activity budgets do require being combined with an estimate of energetic expenditure, so taking the activity budget, how long an animal did something, with how much energy that specific activity took, whether that is respiration rate or speed, or like Kate talked about, using three-dimensional acceleration and ODBA type things. It's using both of those kind of together. So the question that I want to chat with you about for my remaining 3 minutes probably is, can we measure breathing rate and behavior on killer whales to help estimate activity-specific metabolic rate? This data was collected using drones, and this is just a photograph showing Keith, the drone pilot, and Taryn, who's a master's student here at UBC. It looks like she's catching the drone after it went out on a successful deployment. We collected 4 main types of data on this project: the drone video measuring behavior at the surface, the CATS data logger tags. These are the same suction cup tags that Kate talked to you about previously. They measure a whole suite of variables, which we'll go into detail next. One of them does include the animal-borne video here in the lower right. And we also used boat focal follows to track the animal while it was tagged and confirm identification of the individual. These are the 2 variables that I want to look at for the rest of the talk. The drone video captured video while the animal was at the surface, and we could visually count respiration rate, or the number of breaths per surface interval, and quantify their surface behavior into categories. The data loggers measured exactly the same things that Kate had just told you about: 3D acceleration, video, hydrophone, and also a time depth recorder to give us dive behavior. And so we were able to quantify the animal's behavior from above with the drone and couple that with their behavior underneath the surface from the data logger, so we can overall get a more complex or complete picture of what is going on. So we used the drones to categorize the behavior on the resident killer whales. This analysis was performed by Tess McRae, who is a master's student here at UBC. She watched many, many hours of video and categorized their behavior into these 4 mutually exclusive categories. I'm going to show you a video now from the drone's perspective. And to help you orientate yourself, you will see a video of multiple killer whales, but when we are analyzing this data, we're only looking at the 1 tagged whale at a time. So we're coupling down to the nearest second what is happening on the tag with what is happening on the drone. So this illustrates logging. The animals are passively drifting at the surface. If you ignore the ones in kind of the lower right, they aren't really making any forward movement. Compared to when the animals are resting, they have more movement than when they're logging. They're swimming very slowly, but there's no progress in a single direction. With traveling, you can tell visually the animals are moving faster in one direction. They're making definite progress. But again, they're still quite shallow diving. The animals aren't disappearing from view completely. And then the last behavior is foraging. You can see a killer whale here foraging. They're often observed foraging alone or sometimes accompanied by the dolphins here. You can see ahead of them. And we can tell that this is a deeper dive because then when the animal is submerged, they can disappear completely from view. So that's just a sample of some of the really cool videos we got to look at to quantify behavior. Looking at insights so far from the video, this is an example of an activity budget we have on about 500 dives from 8 resident killer whales. You can see the majority of their time was spent traveling in blue, and then approximately equal amounts of time the animals were resting or foraging, and logging, where they're passively drifting at the surface, was rare in this activity budget. Looking at respiration rate so far, the killer whales breathe once per surface interval during foraging, resting, and traveling, but they breathe more frequently when they're logging at the surface. We observe 2 to 3 breaths per surface interval. So why should you care how often this whale is breathing at the surface? Big deal, right? Well, it's important because this shows that previously we have been assuming that 1 breath equals 1 surface interval. So then you can just count the number of surface intervals on a simple TDR and get breathing rate. And that had been observed by a lot of people in the field, and we just quantified and said, yes, that is what happens most of the time unless they're logging, and logging is less than 2% of the time. So that's really promising news. And then coupling this respiration date with behavioral data from the drones can help us improve more accurate measures of activity-specific metabolic rate. I'd like to thank my co-authors as well as the people listed here and the crew that helped collect this data. And if you have any questions, I'm more than happy to chat with you during the break or at lunch. Thank you.
Okay, just one sec. Come on down, Burek. We're going to get you mic'd up. And we do have a question for you, Beth. Right here.
Yeah, just really easy. Is this northern residents?
This is a combination of northern and southern residents based on the tagging data. I see one up on the top right. Hi, Anna.
I'm just really interested in the different behavioral states and if you have any insight into whether it relates to different habitat types.
The question was if any of the behavioral states relate to habitat types. Maybe, I'm not analyzing that for this part of the project, but it's part of a much bigger project, and we can talk about that at coffee. Maybe after you hear Barack's talk, that might kind of help answer some of that. But that's definitely important to take into account habitat type as well. So thank you very much.
Thank you, Beth. Are we set to go? Our next speaker is Burek Sagili. Over to you.
Hello, everyone. My name is Burak Saygılı. I'm a postdoctoral researcher at UBC. I'm using hydroacoustic to solve a mystery of killer whales. Today, I will talk about or research about that. Southern resident killer whale population has trouble, but northern resident killer whale population is well. The hypothesis is SRKW are declining because they have fewer salmon available to compare to NRKW. However, data are lacking on the availability of their prey in the hotspot feeding areas. Therefore, we performed the acoustic surveys at this area to determine density of the Chinook salmon to compare prey availability. Acoustic data were collected from 28 hotspot feeding areas in the summer 2020. We collected this data in the summer because in this time, high numbers of Chinook salmon are returning to their natal rivers. Scientific echo sounders used to collect acoustic data more than 600 kilometers set out, and more than 100 hours acoustic data collected. We use this transducer to collect the data. The recorded acoustic data look like this on the PC. This is the echogram, which is used to visualize the acoustic data. These lines are fish tracks. The scale shows the size of echo from strong to weak. Here is the sea bottom. We had many fish tracks, but only a few of them Chinook salmon, and they were usually near the bottom. These points are surveyed at the northern areas. This scale shows the number of fish per kilometer square. What we found, the densities are very small in these areas. What we found in the southern areas, the densities are high, mouth of the Yuan-Fuka Strait. In conclusion, prey density were higher for SRKW than NRKW in 2020. Prey density were highest on the Switchshore bank. Higher densities were usually close to the bottom. Fisheries acoustics techniques are an effective tool to assess the distribution and density of the prey, and also it can be collect the continuous abundance of Chinook salmon available to SRKW. Thanks to everyone who contributed this study. Please visit our website and contact us. Thank you for your concentration.
Thank you.
First of all, we use the regression from the empirical regression, and then we use the previous data already performed this area, and we compare their size of echo, and usually their length is the main proof of our research. Larger one. We prefer to process first the larger one. Yes.
Yeah, thank you, Barak. And I should also point out on that question is that the previous year we had May Sato here, and May worked with some of the sport fishing guides and was validating, and it does come down to them being so big. And so we're looking at the really big fish. Next speaker is Taryn Scarf.
Awesome. All right. Hi, everyone. As Andrew said, my name is Taryn. And today I'm going to be talking to you about the coastal transient killer whales and their abundance and distribution off the BC coast. So the last few presentations have focused on the southern residents. We're going to look at a different population, the less studied transient killer whales. So I'm sure many of you in the audience today know a lot about transients already, but for those of you who may not, transient killer whales are also known as Bigg's killer whales, and they are the mammal-eating population, whereas the residents are the fish-eating population. So transient killer whales range from northern California to southeastern Alaska. However, for my research project, I'm only looking at a smaller subset of this larger region, the inside waters of Vancouver Island, which is the area highlighted within that yellow circle on the map on the screen.
So I chose the inside waters of Vancouver Island because throughout the past several decades, transient killer whales' presence in this area has significantly increased, and their populations have increased as well. So sightings in the inside waters of Vancouver Island, and particularly the Salish Sea, have gone from rare, infrequent encounters to pretty much daily encounters now. However, how transient killer whales use this area and the seasonal importance of particular regions within the inside waters remains relatively unstudied. So that's what led me to my overarching research question of really how important are the inside waters of Vancouver Island to the transient population. To answer this, I collected sighting reports from researchers, whale watching companies, and publicly available databases from 2016 to 2020. In total, there are about 10,000 sightings, but that does mean that there were multiple sightings of the same group in one day. So to account for this, I separated all of the sightings into occurrences. So if one group, the same group, was seen multiple times in one day, that would only count as one single occurrence. If they were seen with a different group that had not yet been seen in that day, that would count as a second separate occurrence. So you can still have multiple occurrences per day, you're just not getting the repeat encounters of the same groups. So within the inside waters, there are two larger sub-areas that I looked at. The first and northernmost is the North Island waters, which is highlighted in blue. The second is the southern Salish Sea. So some preliminary results from my study, firstly looking at the monthly distribution. So on the left-hand side of the screen is the total number of occurrences per year from 2016 to 2020. So from those approximately 10,000 sightings, it was whittled down into about 3,600 occurrences. An average between the 5 years was about 750 occurrences per year. For the graph that I'm going to show, the x-axis is the month, the y-axis is the total number of sightings or occurrences in that month. And looking at the data from 2016 to 2020, we see that transient killer whales are present in the inside waters year-round. There are some seasonal peaks, particularly in the summer months. The one thing that I do want to note for this project is that, and this graph in particular, is that the data are not yet effort corrected. So that's something that I'm currently working on through analysis. And what I mean by that is we have so many sightings, particularly in summer months, that doesn't necessarily mean that there's so many more whales here. We just have a lot of eyes in the summer compared to the winter. So that's something that I'm accounting for through statistical analysis. But with the effort correction, I do expect to see similar peaks. The first one happening late spring, early summer. The second later in the summer, August and September, which coincides with the influx of sea lions we have to the area, as well as the harbor seal pupping season. So the second graph is looking at the proportion of the population that was actually observed in the area. So from 2016 to 2020, the population present in the inside waters was pretty stable at about 70%. So overall, about 70% of the entire transient killer whale population was present in the inside waters, and that's about 240 individuals out of the, on average, 350. So a really high amount of the population is spending time here. So in conclusion, the inside waters of Vancouver Island should be considered critical habitat for the coastal transient killer whale population. This is because they're present in the area year-round, and we know that at least 70% of their population is using the area. It also highlights how important the prey in this area may be for their population, and it shows that we have a need to assess other key feeding areas within that larger range from Northern California to southeastern Alaska to look at where they're spending their time and potential prey implications in those areas. So I just want to end by thanking all of the individuals and organizations on the screen here for all of their data contribution to the project as well as their guidance throughout the past several years. And thank you for listening.
And thank you, Taryn. Have we got any questions? We have a question over here. I'm going to hand you the mic, Taryn.
When you say that 70%
So it's 70% of the entire population. Yeah.
Thank you.
Do you think that, and again, this might be part of when you correct for effort, do you think, Taryn, reduced effort because of COVID and there were less people on the water? Or I was kind of surprised that 2020 wasn't way less than 2019.
Yeah, so the question is asking about the number of sightings in 2020, if COVID affected that. It definitely affected the whale watching numbers because they weren't able to get started as early in the year. But there were actually a lot of people out on the water. So since we were all stuck at home, if you had a boat, I'm sure you spent time on the water. So there were still quite a few sightings happening, just fewer from the whale watching companies until later in the summer.
Okay, people leave this. I think this is better.
Okay. Thanks, Terry. And our next speaker is Josh McInnes.
There we go. This thing, oh, it worked, okay. Hello, I am Josh McInnes. I'm a graduate student at the University of British Columbia, working with Andrew Treitz. But first off, I'd like to say that this study is a collaborative effort by numerous individuals, a long-term behavioral study of transient killer whales off the California coast. We've kind of got some new findings that are pretty interesting, and I'll share a little bit with you. So first, why study behavior? Behavior can provide invaluable insights into interactions between prey and predators and how predators might be affecting prey populations. In particular, in an area of the California coast where we don't know a lot about how predators like killer whales affect prey populations, this was kind of an area we wanted to really look into to see, in particular with the gray whales, which was mentioned earlier by Stephen Rafferty. This is kind of an important area to figure out how killer whales may be affecting these populations. So the objectives of our study was to provide information on the predatory role of transient killer whales in Monterey Bay, California. This includes defining foraging behavior in prey species, to develop an activity budget, which Beth mentioned earlier for the resident killer whales. We did a similar approach with the transients, and to provide information for ecological models that can be tested on physiology and habitat use. And also to provide information on the seasonal occurrence, as well as prey species and habitat use for that region. So our primary area was Monterey Bay. It's a large bay open to the open Pacific Ocean that is off the central California coast. And it's a pretty special area, being that it has a large submarine canyon that bisects the bay. And it's an area where nutrients from upwelling as well as the California current extend south. Our methods of observation were opportunistic sighting, opportunistic encounters with killer whales through surveys. Through collaboration with NOAA's Southwest Fisheries Science Center, we did survey work, and they collected a lot of information that were part of the study, as well as the whale watching community in Monterey Bay supplied a lot of sightings for our seasonal analysis. With the group, we conducted group focal follows from about 2006 to 2018, where we'd go out every day and we would follow killer whales for between an hour to when sun goes down. And we'd collect information within 50 meters of the whales on their behavior when it comes to breathing, their predation, looking at the movements of the animals, how far they are from each other. We collected different variables to try to define activity states on these different animals here, including underwater observations. Prey species identification was a big one. So a lot of it was, especially in the spring months, which I'll share a little bit about in a couple minutes, but gray whale predation was quite significant in the area. But what was really interesting was defining prey by observation of active predation, where killer whales were involved with the species, but also looking at the quantity of blubber that was in the water. You can see this is a killer whale up in the top here. That's not, oh, there we go. That is feeding on a chunk of gray whale here, as well as the bottom here, and we collect data in samples from prey as well to analyze. The results of our study, though, we had 260 sightings of transient killer whales that we confirmed through photo identification, where we knew who the individual whales were from a previous study that we published, as well as 100 encounters with transient killer whales. And what we found, though, was the majority of sightings occurred in this heat map here from my colleague Kevin Lester, who we were able to look at the highest area where transient killer whales were found in the bay, and we were able to define some of the behaviors with that. To look at the activity budgets, though, we wanted to also get a visual. So the data that we collected through following the killer whales and defining those activities we're using GIS. So you can see here, ArcGIS Pro, we plotted the data for how the killer whales were moving based on increments every couple of minutes. We'd take a latitude and longitude, and then we'd plot it, and we were within 30 meters, within 50 meters to 30 meters of the whales at all times during these observations. So we were able to define some interesting behaviors. One, which was really fascinating, is that a behavior called shelf break / canyon foraging, which is really interesting. It's in the bottom right here, you can see, the bottom right panel, where the killer whales actually follow the canyon within 100 to 200 meter isobath, and they look for prey along that area. And often it's synchronized dives where they're together less than 2 or 3 body lengths apart, and most predations involve gray whale calves when they're using this behavior. Up at the top here is traveling. That was another one in the left corner. We also had open water foraging, which we see in transient killer whales typically up here as well in British Columbia and Washington, where there's a lot of zigzagging back and forth, long dives, sometimes separated by a kilometer between individuals. And then at the top here on the far right, we also had prey pursuit as well as feeding behavior. And the top right panel here was where they are in one location actively feeding, and feeding can last anywhere between a few minutes to over 6 hours. Especially with gray whale predations, the group will stay in one area and feed on a calf for extended periods of time. We also had prey pursuit, and then traveling, socializing, and resting. And here's the activity budget we came up with. We found that shelf break / canyon foraging was the majority of the predation, the majority of the activity budget. Same with open water foraging, and then followed by a few others. Diet was presented with a few different species, but the most dominant in the spring was California sea lion at 33% and gray whale calves at 30%. But you can see throughout the rest of the year, there was a dominate prey was 87% was California sea lion. And this was based on 80 observations we made of different prey species. Seasonality as well, you can see here it was we had 2 peaks, and we did a similar thing to what Taryn described earlier using occurrence, because transient killer whales in this area spend at least 4 days in the region is what we came up with, and if there was an occurrence would be based off that 4 days. So if they disappeared after 4 days, we called that an occurrence. Classified as one occurrence. And then finally, working with NOAA Southwest Fisheries Science Center, we recently have been looking at the gray whale calf count numbers, and we found some significant results with killer whales showing up in the spring months and between March and May, when the gray whale calves are migrating up the coast, and we've observed over 24 predation events. And finally, we just published a new book that's coming out in the next month through NOAA's Southwest Fisheries Science Center, and it has an updated catalog for the killer whales off California and Oregon, as well as information on the studies that we're conducting on the behavior. I'd like to thank my co-authors and people at UBC and NOAA's National Marine Fisheries Service who've been instrumental in this study. And thank you.
Thank you, Josh. We're going to take that off you, and we're going to have 2 questions. So first over here.
Hi there, great talk. Just wondering, why do you think they're using the isoplast to, the isoplast forage in Monterey Bay?
So the question was, why do the killer whales use that particular isoplast, that area along the canyon to hunt? It's really interesting. We see certain groups tend to use this canyon area more often than others, and it likely is because gray whales do cut across the canyon. The initial counts of gray whales when they were watching them migrate up the coast is that they hug the shoreline along this area called Point Lobos, which is just outside of Monterey, and they kind of go right in and then across the canyon. And killer whales use that, transients in particular, use that area to attack gray whale calves. But we're also seeing other canyons. We've had sightings from whale watchers. We've teamed up with the tuna fishermen off the Oregon coast, as well as pelagic birders who have sent in historical data. And we matched killer whales throughout Oregon and California into different canyon systems, which is kind of the next step.
One last question.
Yeah. You said feeding takes a lot of time, presumably when it's a big prey like gray whales. But do groups specialize? So if you look at a group of killer whales that only feeds on dolphins or something, then it would completely change the foraging versus feeding ratio, right?
So the question was, thank you for the question. The question was, do some of these groups of killer whales specialize in particular prey than others, especially because feeding takes up quite a bit of time for certain groups and maybe not for others. During the spring months, we see groups of up to 24, and typically that's quite a few of these animals in these groups actually form the majority of the match lines that we see in the region. We think there's a few groups that use Monterey in the spring more than others, and that's something we're focusing on right now. Thank you very much.
Thank you, Josh.
Hello, my name is Kevin Lester, and I'm going to be talking about geographical information systems applications in marine mammalogy. GIS is, like I just said, geographical information systems, but you can think of it as just fancy math-informed maps. You saw a lot of my work in Josh's presentation already, but I'm just going to talk about like a general applications GIS. So way back in undergrad, I was studying painting at Pacific Lutheran University, and I had the wonderful opportunity to study in Antarctica via a literature program. So I was studying poems in Antarctica, and I was blown away by the marine mammal scene there, to the point where it just became an obsession. And when I got into my master's at the Evergreen State College, I wanted to take the information I was learning, particularly the new skills in GIS, to Antarctica. But there's kind of a funding barrier to get to the South Pole. So I thought of the best local proxy was harbor seals. So I got to work thinking about how I was going to design a harbor seal survey, because at the Evergreen State College you can basically choose whatever you want to study, and as long as you create a cool thesis, then it's good to go. So I ended up salvaging a sailboat that was leaking out the top and the bottom to serve as a research vessel, and I spent a year living on it a couple weeks at a time, surveying the South Puget Sound based on Antarctic survey methods that I had read about, preparing myself for Antarctic pinniped surveys. So at Evergreen in the GIS courses, I learned about how to create apps and use ArcGIS Pro. So I designed my own mobile app to collect environmental information, sighting data, all that, all by myself, out on the water. I had volunteers steering the boat while I looked for subjects, observations, and the boat was outfitted with environmental readers, so that plugged right into the data. And yeah, at the deeper areas, I surveyed via the sailboat, and then in the shallower areas, I got out and surveyed via kayak, going about the same speed. So when you think of GIS, you think of like these big, beautiful maps, nice cartography and everything, but behind all that visual is all this statistical information. So in the bottom, you can see all the environmental data being graphed out. I took information on temperature, on human activity, and where the seals were being found. And in the 3 top maps, you can see the density of the seals based on the season, summer, fall, and winter. And that kind of lines up with their life cycle with molting, pupping, and foraging. Particularly in winter, you can see in that central right-hand side, there's a lot of density out in that large area. They're out there foraging with California sea lions and actually humpback whales and dolphins. I'm surprised to see them out there in the South Sound. It's kind of way down in the Puget Sound, but they make it down there. And I found that the distribution of harbor seals was by season most associated with depth in water count. So from that kind of that baseline, I met up with Josh, and we started looking at more temporal things. So we would collect data on killer whales off the coast of Monterey, and using multiple observations of the same individual, we're able to map out kind of what he was talking about, these different foraging types, and start building a biogeographic model of how they interact with the space that they live in. And moving into the future, we can use drones and sonar to build biogeographic models in remote areas that might not have very good data, because we're kind of spoiled in North America, where we have like all this government data that we're able to just plug and play with our data. But in more remote areas like the Antarctic islands, or you'll have to go out and collect your own and start building this high-res 3D image of what's going on and how the animals are interacting with that particular environment. So that's where we're moving off to. So I just want to thank Josh for bringing me on and inviting me out, and the Oceanic Ecology Research Group for also accepting me and letting me do their GIS.
Thanks, and Bob, you're coming down to get mic'd up. Do you have any questions for Kevin? Kim, do you have any, other marine mammal projects? Are you looking to take on more work? It's quite remarkable. I think I was just blown away by seeing the way you can bring data alive. Or you have to give up your other mic.
Yeah, no, I'm definitely taking on projects. I live for this stuff. If you need a GIS, if you have a GIS need, if you need to model things in space, I am the guy.
Yeah, although I might be first in line, I can tell. Yeah, really impressive, Kevin. Thanks so much. All set, Bob?
Yeah.
There we go. The floor is yours.
Oh, look, there's sound. I feel like I'm on TV or something, and maybe I kind of am. So my name is Bob Sierri. I'm a new postdoc at UBC, and I'm kind of also new to the marine mammal world. I'm excited to be here and most excited to meet all of you. I'm relatively new to this field. I'm just going to describe what I'm going to do with my postdoc project. Just getting started, but it's really fun. So I'm interested, as you might be able to tell from the title, in lung structure and function and pulmonary air patterns in marine mammals. So I'm a comparative physiologist, biomechanist, interested in how organ systems work, and have adapted to biological challenges and evolutionary pressures. And if you're interested in lungs or I guess anything else, whales are a great thing to study because blue whales and stuff are big. The whale lung itself is really big, and it's adapted to life in the water. And what I think is really interesting is how quickly they ventilate despite being so large. And there's a kind of a cool, and easy to use, well, easy to study gradient in adaptation between true cetaceans, pinnipeds. I'm going to be working right now with the cetaceans. So I'm teamed up here with Robert Chadwick at the University of BC. On the bottom with a big fin whale lung. So you can see they're pretty big and kind of hard to see through. And he had a student, Marina Piscatelli, who helped, or did a lot of the work also. So that's she there. You might have met her. I never have, unfortunately, with a beluga whale lung. So I have this treasure trove of data that I can really get to work on and try to figure out what's going on. So again, two things that make the marine mammals really interesting in terms of lungs are the overall size. So how does a lung that big work when it has the same basic design as the lung in our bodies, or the lung in a shrew or something? And how do they deal with this being in the water thing? So the first thing is, although sometimes they do log versus take shallow breaths, marine mammals breathe really rapidly at the surface. This killer whale comes up and breathes in and out really fast, and that's a lot of air going through. And so the airways, the upper airways in these animals, as well as the kind of the major airways in the lungs, are really reinforced. And if you think about a garden hose versus a really enforced high-pressure hose, that's kind of what we're dealing with. And the shape of the lung itself and the branching pattern of the airways has adapted to that. So on the top, there's a lung rendering of a CT scan from a minke whale. And if you compare that to a human lung, there's not really that many lobes in there. There's just the one lobe, and there seems to be a quite big, high diameter main bronchus that leads to several other branching ones. So how might the air move around differently in that lung versus our lung has not really been studied yet, because it's difficult. So a little bit more about that, that elaboration. Here's an image of a dissected primary bronchus from some of these animals. You can see these really thick
tracheal rings. And on the left is a cladogram looking at the differences in marine mammal versus terrestrial mammal lungs. And you see there's these myoelastic sphincters, so heavy, smooth muscle elaboration all the way down to almost to the gas exchanging regions, and a lot of tracheal rings. And in the pinnipeds, there's kind of more robust walls around different parts of the alveoli. And the bottom left is actually a slice through a bottlenose whale lung, and you can see moving laterally is that primary airway, and along there are these myoelastic sphincters with heavy connective tissue. So what is this about? The hypothesis is that because they're breathing so rapidly, if you try to expand and contract the lung so fast, you'd have pockets of the lung that got pinched off or air was trapped in. You couldn't have a full exhalation and inhalation. But if you have this elaboration of these connective tissues, and they essentially can keep that airway patent during a forceful exhalation inhalation. But we're not quite sure how the air will move around because it's hard to measure these things. And I'll get to that in a second. And the second thing I want to talk about is obviously marine mammals dive, and when they do that, the lungs are subject to external pressure, and they collapse nearly completely and then successfully reopen at the surface. And in order to do that, you can't have air trapped in the small passages. You might have barotrauma in the lung. So the lung has to be designed to be protected from barotrauma. The spongy tissues have to have the air move into the bigger airways where they cannot be, they don't get as collapsed. So how might lung physiology and biomechanics have adapted to these pressures? The best way to study this, I think, is using computer tomography and computational fluid dynamics. And we can try to ask some questions along some ecological gradients. So if we compare an orca and a beluga and a cuvier beaked whale, they're all odontocetes, but they differ in how deep they commonly dive and how fast they commonly ventilate. So if we compare the anatomy between these three species and then also the functional airflow patterns, that's probably the best way to get started. So the anatomical comparisons can be made with something like this. This is just a quick rendering, a volume rendering of the scan. You can see there are big differences, but in order to make quantitative comparisons or real better comparisons, we need to do a slightly more advanced technique, and that is computational fluid dynamics modeling. So the way this works is we take the CT scan as the raw data, and we're going through from the top to the bottom of a minke whale lung, and you see the big airways start to show up. And what we can then do is segment the regions of interest out of that scan. So in purple here, I've segmented some of the airways in this thing, and we'll turn that into a 3D model, fill it with a computer mesh, and then have a supercomputer run the Navier-Stokes equation in there, given some boundary conditions to try to compute how air is flowing through the lung under different conditions. As the lung is filling, the lung is emptying, or as pressure is being applied to different parts of the lung, perhaps during a dive. And then the last thing I wanted to point out is in these lungs, you can really see the big airways, but the branching pattern goes down to a greater degree of a smaller size than we can visualize. So what we'll have to do is I've teamed up with a team in New Zealand that does this with human lungs, and they fill the difference between the surface that we can measure in the CT scan and the actual surface of the lung with a discretized computer model based on the branching angle and relative size of these things. So we're going to match the anatomy we can measure with a computer-generated model to make our other computer-generated model. Okay, so I don't have any really results to show you yet, but I just wanted to show this video to give you a sense of how these lungs work. So this is a minke whale lung that I've been processing. This is just the lung rendered surface. You can see the outside of the lung. You can see the major airways and leading to the smaller airways. Again, it's just a single lobe with a pretty consistent branching pattern, which is unique compared to terrestrial mammals. And then in a second, I'll turn on part of the rough segmentation of the airways. So this is the negative space that the air is in, and that's going to be essentially the main part of the computer model. So, and then the last thing I wanted to show is just what kind of results you might expect. So this is a similar project in my PhD using the same technique on lizard lungs. So this is just showing you the kind of data you might get to see here in a few years if I pull this off. And these are streamlines put into the CFD model to show how air is flowing during inspiration and expiration. And this is beside the point, but we could use it to ask a question about a hypothesis about how the bird lung may have evolved and what it had to do with anatomy. So with that, I'm happy to take questions.
Maybe I can ask just one question before I take a break, which is, What should a naturalist on board a whale watching boat tell their passengers about your research?
Yeah,
what should
that's
they
a
know
great
about
question.
the lung that they could pass on that would just amaze the guest?
I would just say if you every time you watch one of these whales breathe, the lung is the size of you or bigger, and the air is going in and out really quickly. And also that whale pretty much spends most of its time holding its breath. You think about take a breath and hold it. Maybe you can make it a couple minutes, but one of these whales might die for half an hour and breathe for a few seconds at the surface. So it's almost like they have a really backwards way of breathing compared to us in terms of breathing versus apnea.
Yeah, very cool. Thank you so much. We're going to take a 15-minute break, and we'll be back here. Anna Hall will be first up. You don't want to miss it. Bob, that was great. Yeah, you're a very good communicator, and I just knew we had to connect beyond your physiology.
No, I agree.
Okay. Welcome back, everybody. I'm not sure if my mic is on. If I'm muted, perhaps. Check. Am I muted on here? I'm on now. Check. I'm not sure. There it is. Now I can hear myself with lots of reverb. Okay, welcome back. Due to our technical challenges, we started 15 minutes early, which means we will be cutting 15 minutes into our lunch break. So it'll be a shorter lunch than previewed. Earlier we heard about how we need to think about the lungs of large whales about the size of a person. Well, I think you could also think of lungs of large whales of the size of a super small cetacean, porpoises. So Anna Hall is our next speaker.
Thank you, Andrew. Good morning, everybody. I'm here to talk to you. There we go. Here to talk to you about the super small cetaceans and the medium-sized superstars. And so, because there's a timer on with 5 minutes, ladies and gentlemen, fasten your seatbelts, please. We are going to circumnavigate the North Pacific Ocean in the next 4 minutes, 56 seconds. Okay. So, starting at the United Nations, why not? There's a big push around the world to reach these sustainable development goals. And the one that a lot of my work falls into is Sustainable Development Goal number 14, obviously with life below the water. And there's a lot of components to this, but in a nutshell, it's about conservation and it's about sustainable use of resources within the ocean. And these are two very, very important pieces, whether we're talking about small cetaceans or medium-sized cetaceans or even large cetaceans. I spend a great deal of time at sea, and I spend a lot of time watching animals, trying to decide what it is that they're telling me. And not because we're trying to, communicate telepathically with them, but what they're telling me about their needs, their ecological and their habitat needs. And that's based on where they are, how many of them are in the group, how many groups there are there any communications that are going on between the groups? What types of behaviors do we see? And what types of responses do we see from perhaps human activities or even other animals' activities? So, I always try to think about what is it that these animals are trying to tell us? Oh, we've got a little bit of something happened to the formatting there. Let's see what's on the next one. Yeah, get it right. We do try. And I suspect that these animals are terribly polite. But in all seriousness, I spent a huge amount of time trying to decide what are the animals telling us? What do they need from us as people? Because we've had a huge influence on our oceans. Back in around 1900, it was said in England, as I've mentioned before, that harbor porpoise were seen to occupy knee-deep waters, and mothers would bring their calves into these locations. So that has me thinking, here, what has changed? Because, we can't go back in time, but we can look at maps. And just as an example, this is the Victoria Harbor. It was mapped in 1853. Here's our Google rendition from 2014. It's profoundly different. We can see that there's the mouse. We can see that the islands in the middle, in the mid-1800s, hardly exist. We can see that this very complicated shoreline has been smoothed and extended. The harbor here, which used to extend into mudflats, is now greatly shortened. The width between these two pieces of land is much narrower than it was. And again, we see that the shoreline is far less complicated. We also know that there are new structures. For instance, these berths and the breakwater that simply didn't exist less than 200 years ago. Now, I can't tell you what harbor porpoise were doing in this region 200 years ago, but given the name harbor porpoise, I would say that we've got a pretty high likelihood that they were in this region. We also know that in the 1950s, they were able to be seen from shore in the Victoria region, and these days you cannot. You must go out at least 5 miles. So we've had some big changes. But we also know that these changes are limited in space and time. And there are regions on a BC coast where harbor porpoise can still be seen in the nearshore habitat. You can see the grass in the foreground here, and this is research done by the Porpoise Conservation Society. We have 2 land-based sites, one Queen Charlotte Channel and the other at Saturna Island. And we have determined that we have interannual occurrence of harbor porpoise moving into these shallow habitats. We're seeing a number of different behaviors, including mating, foraging, calves, and occasionally they do poke their little heads above the water. And so we get to have a better look at these super small cetaceans. So transiting across the North Pacific to China, where there is another super small cetacean that is in trouble, but we have a vastly different conservation program going. This is the finless porpoise, Yangtze finless porpoise. There is a captive breeding program with fantastic research facilities and enormous oxbow lakes that have got nurseries and medical facilities. And the scale of these oxbow lakes is very difficult to convey, but all I can say is when I'm out in Juan de Fuca Strait and I look down the channel, it reminds me of the reserves that are set up for Yangtze finless porpoise. In 2021, the Chinese government enacted a 10-year fishing ban so that the Yangtze finless porpoise is not competing for fish with humans. They can hopefully rebound their population a little bit, be taken from these reserves into the Yangtze River and perhaps repopulate. China admitted that they failed Baiji, which was determined to be extinct in around 2007, very sadly, but they are doing their very best not to fail Yangtze finless porpoise. And you can see here, this is the shore of the Yangtze River. It is vastly changed, but China is taking some very tremendous steps to try and save this species. And they have had successful captive breeding with calves born successfully since 2005. So there are some real amazing conservation actions taking place. COVID has unfortunately, of course, changed our world, and we haven't been able to get back to Wuhan, where the captive breeding facility is, but we're certainly trying to provide as much support externally as possible. Interestingly, in China, Yangtze finless porpoise, known as the smiling angels of the Yangtze River, good media spin, they are reported regularly in the media. Lucky for Yangtze finless porpoise, they do not have any medium-sized media superstars living nearby, as we do here in British Columbia. The work here on killer whales is based on research, monitoring, mitigation, repeat. Last year, I spent 70 days at sea with the Department of National Defense. I've been working with them since 2017, doing just this, research, monitoring, and mitigation, helped develop the Marine Mammal Observation Communications Protocol, which is based on in situ monitoring and real-time communication of the presence of cetaceans within 5 different ranges. And last year alone, we did 1,300 nautical miles at sea with over 380 hours dedicated to the active watch during the operations. We look for these medium-sized cetaceans, but also for the super small, which means we have to have good sea conditions. And when we don't, we are able to relay that information directly to those who are enacting the operations by the Department of National Defense. We've also had the pleasure of working with the Pachirat First Nation in Western and Central Juan de Fuca Strait on a multi-species research program. This is a collaboration between Pachirat First Nation, SMRU Consulting, Port of Vancouver Echo Program, and CView, which is where I work. Dr. Dom Tollett and I developed this line transect survey, which extends through Central and Western Juan de Fuca Strait. And just very quickly, you can see here lots of purple humpbacks, a few of those medium-sized cetaceans in red, but very importantly, those glowing yellow sunshine orbs of harbor porpoise distributed along the shoreline. Again, moving into those very, very near-shore habitats. SMRU Consulting developed this sightings kernel density map based on 2021, and we can see the hotspots. When we compared it with including the 2020 data, we can see that we get a hotspot out towards the western entrance. So just very quickly to finish up, we've got the International Save the Vaquita Day. We were able to have it again this year, which means vaquita still exist. And it was during this event that I had one of those light bulb moments when Dr. Lorenzo Rojas Bracho was talking, and he was saying one of the fundamental aspects of vaquita conservation and why we have failed this species is because we are not treating this as a financial crime. That got me thinking. All of the work that we've been doing with China and here in British Columbia has been based on collaboration, communication, and trust between all parties because we have only one planet and we are working together, and this triangle works. When it falls apart, it doesn't work. And I can tell you for sure that we are failing vaquita. Looking at these 10 trusts, which I will not read, but just let you go through them yourself, learning from those who have extensive experience with these very complicated conservation situations. The single most important thing that we have to have is trust. And there are these 10 trusts that Jane Goodall and Mark Beckhoff have put out. And we can trust that the nets are still in the Sea of Cortez. We can also trust that the vaquita, based on the last scientific expedition, still have calves. We also know from the scientific research, looking at the genetics, that there is still hope that this population, this species, could come back from very low numbers. It will take a global village to save this species, and given that their range is less than 1/1000th of the Earth's ocean surface, perhaps we can do something just a little bit different so that we don't continue to fail this species. And because they still exist, we are taking a socioeconomic approach, because this is not a science problem. Science has identified the problem, and the tremendous work of many of the scientists in both Mexico and the United States has got us to this point. But we have recognized we have a socioeconomic problem. So we are building a collaborative group between Dr. Samuel Turvey at the Zoological Society of London, Dr. Tom Jefferson at Viva Vaquita, and those of us at the Porpoise Conservation Society to try a new approach, which is multidisciplinary, that will take place in the immediate, medium, and long-term timeframes. But this really is our last chance. We are down to approximately 10 to 20 animals for the entire population. So I would say, many people would say, well, these are super small cetaceans. I would perhaps call them super small cetaceans, because they really are. We do have these medium-sized superstars. They're all important, all definitely part of our ecosystem. And if we look at the United Nations, life below water, we have to conserve and we have to sustainably use, because for our future explorers, is this really what we want them to see? Or would we rather that they see vaquita? And just in closing, I thought it was interesting that this book is closing with a coda, which those of us in marine mammal biology and acoustics think of Watkins and Shovel 1977 using a coda to describe the sperm whale clicks. But it is, in fact, also the last piece, the last event can be used in closing. And this book closes with, after all is said and done, science is betrayal. So we continue to spread the word about these super small cetaceans. The harbor porpoise, you can purchase this new book. It's on Amazon. And that is Jack from the Vancouver Aquarium, gracing the front cover. Of course, we always have to say, adopt a porpoise, because you too can live your life with a porpoise. I'm not sure what's happened to the fonts there. And all of this is collaborative. Thank you very much. And thank you to all of you.
Questions while we're setting up for the next speaker?
Are there any indications main vaquita are managing to learn to avoid nets at all?
Yes, so that's an excellent question. And in fact, the answer is yes. I've had many people say to me, just give up. This is a doomed species. Put your efforts elsewhere. But what we do know about the vaquita that we have seen in the Sea of Cortez is some of them do have net entanglement marks, and they have survived, which means we have the survivors. And so our work is continuing for those individuals.
That's going to be it. Thanks, Anna. And I think we're set. Amy Mignot.
Okay, hi everyone. My name is Amy Mignot. I'm a research biologist with Oceanwise, and I'm pleased to be here to represent the authors of the recently published work in Frontiers titled Porpoises After Dark, Seasonal and Deal Patterns in Pacific Harbor Porpoise Aggregations, one of North America's fastest growing ports. So the Pacific harbor porpoise is listed as a species of concern under SARA. However, this is largely due to data deficiencies within the North Pacific. And we find unusually large aggregations of up to 1,000 individuals around the port of Prince Rupert, which we believe to be a potentially important habitat. So considering the high vessel traffic within this area, as well as the proposed port expansion, this could likely disrupt the porpoise activity moving forward. So this study aimed to bridge these knowledge gaps by generating data on deal activity and densities of porpoises around the port of Prince Rupert to identify biotic and or abiotic predictors of porpoise aggregations, and to build a comprehensive picture of porpoise habitat through methods that I'll touch on in a second here to inform the conservation needs of porpoise moving forward. So this study used mixed methods, combined passive acoustic monitoring, mainly C-pods and F-pods, as well as land-based surveys. So the porpoise echolocation clicks were detected using the built-in C-pod and F-pod Kernow classifier. There was some environmental variables used as well, including chlorophyll A, turbidity, sea temperature, and practical salinity, and then generalized additive modeling was used to test the influence that these variables had on the echolocation activity. So here we have a figure of the study site around Tuck Island, and the pod is right there with the 400-meter radius surrounding it. And I do want to draw your attention to the historical sightings, particularly those red triangles, which indicate sightings of 10 plus individuals and the proximity of those to the pod. You'll also see those dashed lines there indicate major shipping lanes, which have consistent traffic throughout the year. So overall, they found that porpoises were detected in 96 of C-pod days, 100% of F-pod days, and 86% of visual surveys. And on the figure on the left there, we have porpoise detection minutes through a 24-hour period, and on the right, a average of the porpoise positive detection minutes over months, so you can get a seasonal view. And what we can see here is that porpoise activity is increased during periods of darkness, as well as a seasonal increase around May to July, as well as a smaller increase in winter months. So, as porpoises feed on small schooling fish, it is likely that these trends, particularly the seasonal trends, relate to seasonal prey fluctuations. As well, the nocturnal behavior seen here could likely be attributed to the fact that herring are much more readily available in this area at night. And here we have more of a breakdown sort of through the months, and I want to point out these spikes during daylight time between January and March. And these likely also relate to local forage fish fluctuations, as sand eel and sand lance are more readily available during daylight hours at this time. So to conclude, porpoises use this area year-round. The patterns do vary seasonally and daily. And we can use these hotspots potentially to inform mariners, industry, recreational boaters of periods of time in which porpoise are most active and abundant, to hopefully potentially mitigate any disturbance that this might have. And next, we'll be looking at analyzing feeding buzzes to determine foraging rates in relation to the presence of vessels. So that'll be an exciting thing coming out soon. And I want to say a special thanks to the groups and people listed here, for which this project wouldn't be possible without. And I'm happy to take any questions right now.
Thank you, Amy. Do we have any questions? Amy, can you tell us about other research that OceanWise is doing with small cetaceans?
Yeah, so we have our ongoing porpoise work. Like I mentioned, we're going to look at foraging. Mainly in the North Coast, which is where I look, we look at humpback whales. I know in the South they're doing a lot of work with killer whales as well, similar to what a lot of you are doing from what I hear with drones and everything. So yeah, I would definitely recommend keeping up to date with OceanWise research. There's a lot of cool stuff coming out.
Thanks very much, Amy. Next up, we have Katie Florko. All set, Katie?
Yes, thank you. Thanks, Andrew. And hi, everyone. My name is Katie. I am a PhD student at UBC. I'm actually going to talk about some work I've done with DFO, though. And I wanted to thank Andrew for inviting me out today, especially because I've never been on a boat for work. And I've also never worked on BC marine mammals, so thanks for having me. So yeah, I'm going to talk about some aerial surveys with DFO in the high Arctic. So this is our study area here, the northern tip of Ellesmere Island in Canada. Not showing. Okay, cool. So this is our study area, the northern tip of Ellesmere Island. we did aerial surveys here, so we were flying transects throughout the Lincoln Sea, Hall Basin, and Archer Fjord. And this was to get an idea of what marine mammals were out here. This is kind of a preliminary study to see what's out here as the area is changing with reduced sea ice. So we were flying transects throughout here. We had a visual observer in the plane that was looking out the window, counting all the marine mammals they could find. We also had a hole in the back of the plane with a mount that had a camera. So if you look over on this mount, there's a camera that's there. And that camera is taking photos every 2 seconds. We're flying at about 1,000 feet, 100 knots, so it works out to about 250 meters wide. And then every photo, since it's every 2 seconds, has about a 30% overlap between photos. So essentially what we get is, you know, an archive of the entire transect line, which is awesome to be able to go back and score photos. The photos looked something like this. There's a bunch of examples of them. So they're really neat and really beautiful, and it's awesome to have them archived, but it's also, there's a couple of problems with them. One of them is that it's really hard to see animals. So as you can imagine, there's nothing in here, I think. Here's an example where there is something. So in that red box, zoomed in, there's a seal. So a couple of problems with that. And then the other big problem is that, you know, we're taking photos every 2 seconds. So this is a 1-week survey. We had about 30,000 photos. So as I mentioned before, I am a grad student, which by definition means I'll do anything to procrastinate my thesis. This is where I draw the line. So luckily we have many tools to help us with this. The one I'm going to talk about today is infrared. So infrared is really useful in wildlife studies. So for example, it's useful if you can't see any visual contrast. So a really good example of how infrared has been used has been for studying the emergence of bats at night. You know, a dark brown bat, dark sky, it's really hard to see. So the idea here was that we would be able to pick up things like white bears on white sea ice. And that's what we found. So luckily, so this is a little bit more detail about the back of the plane, what we had. This is actually, there's actually 2 cameras here in this mount. So we can see that the one that we see here is the infrared, and then on the other side of the mount is the DSLR. So we have both the DSLR going and the infrared at the same time, and we're using them in kind of a complementary way. So they were each hooked up to computers that were archiving all of that footage. So here's an example of what this would look like. So the workflow would typically be that you would go through the infrared and you'd look for hotspots. So here's an example in this white box. There's a hotspot. I'm saying this is all very relative, that this is easier to see than the photos. It is helpful that within the infrared software, there is a histogram of the color, or of the temperatures that we're seeing. So we're able to see kind of spikes of hotspots and then look back at these spots. And then what we do is find the corresponding photograph and the corresponding spot, which is in that little red square, zoom in, and sure enough, there's a polar bear patiently waiting at a seal hole. So pretty neat. Here's another example of some seals. So here's 2 hotspots in this view. Zoom in, and we can see, you know, there's 1 seal along that crack, and then there's 3 seals waiting at a hole. So it's kind of neat to show, I think that this really highlights how these 2 methods are used in a complementary way. So, you know, we can use the infrared to detect where the animals are, and then we can go back to the DSLR photos and identify the species and how many seals there are or whatever is going on. And so this is all things that we kind of expected to see. You know, we expected to see bears on ice or seals on ice and be able to directly observe these animals, but we also indirectly observed some animals. So we saw some narwhal, which was pretty neat. So here's an example of the photograph of the narwhal. So if we're looking at the animal in the rectangle in this photo, and then the first rectangle, the top rectangle in the infrared, you can see that really hot spot that corresponds with the animal, but what we're seeing is this trailing behind the animal in those other rectangles. So that's what we call fluke prints. So each of those mixing of waters is what we're seeing, which represents the last 3 fluke strokes here. So a couple of reasons why we're seeing this. So one of the reasons is because it's a perfectly calm day, of course. And the other reason is because we're having like a thermally stratified water here. So this was in Archer Fjord, which has a lot of glacial runoff. So the water on the top of the surface is colder than the water beneath. And so then when the whale is swimming, it's, you know, kicking up this warmer water. So that's why we're seeing this print. And this is really helpful for our detection methods because, well, first of all, this area that we're seeing the evidence of this animal is a lot larger. So it's a lot larger than just the animal itself. There's this huge trailing. And it also lasts a lot longer. So usually when we're flying aerial surveys, if you want to see a whale, you know, they're only going to be at the surface for 1 or 2 seconds. These fluke prints lasted for the entire duration that we were flying around there. So it was really cool to be able to see those. And I'm glad that we were able to pick up these animals because these narwhal were also turned out to be the northernmost recordings of narwhal. So that was a fun thing on the side. And so that was in Archer Fjord, that little fjord on the top east corner. So pretty neat. So in summary, this 5-minute talk is that infrared is cool. Bring expensive gear to the field, nothing will go wrong. So thank you to collaborators and pilots.
And just while Lisa's coming down to get mic'd, Josh, you have a question?
It might seem like a simple question, but how do you store your data that much?
Yeah, so Josh's question was, how do we store the data? And the answer is gaming computers. Yeah, those are really good at storing data really quickly. Yeah.
Another question?
The field of view of the photos, does that overlap completely with the observers?
That's a good question. Yeah, so the question was, does the field of view of the cameras overlap with the view of the observers? The observers can view more, because they can see more than just 250 meters. It's really hard to limit the observer's view to just 250 meters. It's really hard to be able to assess how big that is. So yeah, there is a little bit more, but still the observers counted a lot less animals than the infrared did, of course, yeah. Yeah.
And the relative heat loss, who's really the leakiest? Is it the seals or the whales? Or
I'm not sure who's the leakiest animal. That's a really nice question, though. One of the things that we did think initially when we saw those fluke prints was that the animals were warming up the water behind them. But I don't think that's the case because these marine mammals are completely evolved to be able to not lose heat in the water. So, yeah.
Thanks so much, Katie.
Do I have to do anything with the timer?
Next up, we have Lisa Walker.
Hello, everyone. My name is Lisa Walker, and I am a whale song theorist. I take my background in music and apply it to understanding the wonderful song of the humpback whale. Humpback song has been called the most complex in the animal world, but really, what is it that makes it so complex? So I have come up with the 6 Ps of humpback song. I'm just going to actually put them all on here for time. Basically, humpback song is composed of patterns, and there's complex patterns, there's compound patterns. These patterns display plasticity in that they change over time. And these changes are progressive, the 3rd P. They result in large-scale changes over decades. But even though the humpback song is consistently changing, there's a persistence to them. There's certain elements that stay the same over time. Each region may have its own song, so the song may be population-based, and it is sung in the context of a polygynous mating context. And so this is not my research. This is research that Jim Darling, Roger Payne, Eduardo Mercado have done it, and so it paints this picture of this ever-changing song with some commonalities across geographies. So just to dive down into the plasticity of it, we have evolutions, yearly evolutions or yearly revolutions, depending on the extent of the change. Each whale can have embellishments to show off their singing prowess. There's also individualization. Each whale might have its own voice. The song changes when they interact, either with boat noise or another singer comes along. And each song begins, or each singer begins the season singing fragments, and then as the season progresses, these fragments grow into song, it becomes more and more complex, and then at the end of the season, it starts to wane. So my goal is to understand the rules and the relationships behind humpback song pattern. And what I realized coming into this field as a musician is some of the scientific tools and techniques may obscure some of the patterns that I've been searching for. The bird song model is fantastic for some aspects of song, but in some aspects it's limited. The speed and the range of the cultural transmission and the pattern complexity and generativity. And so my question has been, well, can we turn to the logic of human music to actually understand humpback song? And so my great quote, this took me years to actually come up with, but the idea is that every piece of human music is a song, but not every song is a piece of music. And when I ask people, well, what do you think is the difference between something like a humpback singing his song and us playing some music? And a lot of people will say, well, how about entrainment? We have a beat to our music. And if I was to play a beat right now, all of our brains would sync up. If we were to play music together, we would actually play it together synchronously. Humpbacks don't sing synchronously. Human music, there's a lot of emotional content to human music. I could play you a sad song and we would all feel sad. I could play you a happy song and energize the room. And there's also this idea about empathy, that if we're listening to music, our threshold boundaries become lower and we will bond with each other when we're listening to music. Now, all of these things may or may not be present in humpback song, which is really thought of still as a stereotype display. We don't really understand yet if there's emotion or empathy in song. So my background is in classical violin, as well as learning African drum rhythms on the djembe. And so these two things have given me an idea of what melodic syntax looks like and what rhythmic syntax looks like. And while I don't have a scientific background, I've teamed up with some fabulous people. One is Lawrence Doyle, who's the principal investigator of a whale SETI project, and Dr. Fred Sharp, who's with the Alaska Whale Foundation. And together we're funded on this project with Templeton Diverse Intelligence Initiative. And so this is giving us the latitude to ask some questions about these whales that aren't usually asked. So what we're doing is we're taking an apples to apples approach to map across the species. And so the first one is, how do we deprovincialize our terminology? How does a scientist and a musician speak to each other about these things? So how do we define pitch? How do we define melody? How do we define phrases? How do we define patterns? And so right now that's what we're doing, is we're unpacking all of the terminology to make sure that it's not human specific or whale specific. Then the next step is to disentangle dimensions. Music is a wonderful array of dimensions. We do have the melody, we have the rhythm, we have different segments of patterns, we have long verses, we have choruses. And so how do we apply different logics to analyzing different aspects of both our species' song? Now with Lawrence Doyle, we are also applying information theoretical techniques to actually understand where the information may lie in the song and what's its communication potential. And we're also adapting tools and techniques from musical composition and analytics, specifically machine learning and artificial intelligence, to actually understand how might we create an algorithm to generate whale song automatically. We're looking at how do we create human music automatically, what are the rules and regulations that go into those algorithms. So our discoveries include the ability to compare patterns to patterns, singers to singers, and song to song. We've been able to isolate different aspects of the song each year and be able to compare it across years and geographies as well. And we're kind of halfway to figuring out a whale song generator. Taking a bunch of different algorithms, we can create different patterns, the logic from, tracing the logic from Hawaii song from 2013 all the way to 2023. This last decade, they've been able to track all the different changes and then apply some mathematical formulas to actually see what humpback song might be like in 2024. We've been looking at dynamic playback methodology. When we're talking about static playback methodology, like just broadcasting underneath the water a specific song or a specific song pattern, it's not as intuitive or it's not as interactive as actually having controls to change what you're broadcasting to the whales. So we're looking at what dynamic playback methodology might look like. And ideally will open up the doors to communication. The focus for me, though, this year, I'm a naturalist with White Rock Sea Tours, is to focus on the local population of humpbacks now in the Salish Sea here. And of pure luck, I was able to record a humpback, a local humpback named Bond. I was able to record his song in Hawaii last year. So when I go back to Hawaii this year, I'm going to look for Bond again. But my quest is to really understand, is there a local Salish Sea song? So thank you.
Do we have any questions? Go ahead. Sorry, let me go over here, if that's okay.
So in terms of frequencies we look at, it's basically the 20 to 20, so all within our hearing range. I do recognize that humpbacks might vocalize lower and higher, but there's so much information within those bands that that's where we're sticking to.
Okay, are we set to go? Very good. Thank you very much. Screen up. And our next speaker is Nikki Diogo.
Hi. The Arctic underwater soundscape is a kaleidoscope of sounds. The sounds of geophony, anthropophony, and biophony change seasonally the underwater sound levels. Within a cacophony of the most fascinating biological and ice sounds, the Arctic soundscape is dominated by the deep moans and the jazzy songs of the bowhead whale. The bowhead whale is a marine Methuselah, and among its many world records, it's also the only endemic mysticid in the Arctic, and it's a species of high nutritional and cultural value for the indigenous communities. Out of the 4 bowhead populations, I focus on the Bering Chukchi-Beaufort Sea stock. Around 16,000 BCB bowheads travel more than 6,000 kilometers every year from the northern Bering Sea to the Beaufort Sea in early spring to feed, and then they return in early fall. Now, a crucial question is how stable can this migration pattern be while the Arctic is warming and the entire ecosystem is changing? Looking for answers, we went to the Amundsen Gulf in the eastern Beaufort Sea to study the acoustic ecology of the bowhead whale. In September of 2018, and for the entire year, we sank acoustic recorders and oceanographic sensors at 3 sites in the southern Amundsen, 2 at Cape Bathurst and 1 off Pierce Point. And we were looking to find out when and where the whales are and why. So the objectives were to quantify the spatio-temporal patterns of the bowhead whale and also merge those with environmental variables in order to get a sense of the conditions that affect the bowhead distribution. We used the Soundtrap 500 to record the ocean sound, and we programmed it to record 5 minutes every hour. Then we ran an automated detector through all our recordings, and then I manually analyzed the 10% of all the recordings and 100% of all the sound files that the detector found bowhead called in order to verify those calls and also record the presence and the number of all bowhead calls in its acoustic file. So the results of this work showed that yes, bowhead whales were present at all our locations, and they were almost continuously there from early spring until early fall, as we thought they would be. But the most exciting result from this analysis, our most cool finding, were those few but clear bowhead calls in the winter months throughout all our recordings. So those represent perhaps very few individuals that they never left their summer foraging areas. Instead, they spent the entire winter of 2019 in the southern Amundsen Gulf when ice was at its peak. But winter of 2019 was a particular one with unusually low sea ice coverage. You can see that here with a red line, and Stephen mentioned that earlier as well. And from our habitat modeling results, we know that ice concentration is the strongest predictor for bowhead presence in this area. And now, from my newest analysis, I can tell you that this overwintering event seems to be, at least for now, not a permanent phenological shift to the bowhead's migration pattern, because I was not able to find any bowhead calls in the analysis of the next year's acoustic recordings from the same area. We used the mixed effect models to quantify the very clear seasonal pattern of the bowhead presence, with higher presence in, from April until September, and overall higher presence at CB50 at all sites throughout the year. And CB50 is our most shallow site. So Cape Bathurst is an area close to a Polinia site, and where the absence of packed ice, or better, the presence of leads of ice throughout the year would allow the animals to spend the winter there. We did not find any consistent diel patterns across sites, but we did find higher detection rates at 5, 9, and 11 in the evening at the deep sites in the summer, and those could be perhaps related to the diel vertical migration of copepods, which is the bowhead's main food item. Our best statistical models showed that the bowhead detections in this area would increase with the decrease of sea ice concentration, wind speed, bottom temperature, and salinity. More or less might have their preferences. And, but we did find a positive relationship with zooplankton density, and that could reveal a clear predator and prey dynamic. The negative relationship with wind speed is because strong winds increase the underwater noise, and that noise can mask bowhead calls. But ice has a dampening effect on noise, so it can decrease that effect in the underwater soundscape. So you can see here that as ice melts, the underwater sound levels immediately rise, and those are largely driven by wind, that yellow line. Now think what can happen to this underwater sound as soon as shipping starts increasing in the area. The more the Arctic becomes ice-free. And with that beautiful thought, I'd like to acknowledge my co-authors and my wonderful supervisors. I'd like to thank the organizers of the symposium for bringing us together. All of you for being here. I'm excited to connect with this beautiful community today. Thank you so much.
Thank you, Nikki. Do we have any questions? Do you want to ask a question as you're getting set up?
I do. That's nice.
Hold that one and you can get mic'd.
Oh, hey. Yeah, I was just wondering what type of detectors you ran given the diversity of bowhead calls.
Yeah, good question, expected from you. We used the JASCO detector, so Xavier helped us with that. And it was only on moans because bowheads make such a variety of calls. And the detector actually did pretty well with the daily presence, but it really underestimated the number of calls that we detected manually because of the loudness of them. It's easier for an analyst to identify them.
Thank you, Nikki. Up next is Caitlin Palmer.
Hi, everyone. Thank you so much for inviting me. Today, I'm going to follow on to Nikki's great talks and all the great acoustics talks today with some practical implications of understanding detection functions, so the probability of detecting a call or an animal as a function of range and various other covariates, and how that relates to conservation. So I'm originally from the East Coast. On the East Coast, we have very good sports ball teams and North Atlantic right whales. Like the southern resident killer whales, right whales are highly endangered. There's about 300 remaining, and the biggest threats to right whale survival are ship strikes and entanglements. And as part of the conservation strategy for North Atlantic right whales, passive acoustics is deeply involved in two ways. First, in long-term monitoring, so understanding habitat distribution. It's shifted rapidly in the last couple of years, as well as behavioral patterns and whatnot. And secondly, we use real-time monitoring to enact real-time conservation movements. So here is a quick image from the Listening for Whales project at Cornell. These are different buoys that are listening for right whales in the Boston shipping lanes. And what's going on here is when a right whale is detected, it lights up on this map, and shipping industries coming through there are asked to slow down, and some that are contracted are required to slow down to reduce the probability of running into a right whale. So this is all well and good. We know now that how important passive acoustics is to this recovery plan, but we've got some basic questions. If you've ever asked a bioacoustician, how far can you hear a whale, you will have gotten the very disappointing answer, well, that depends, because it depends. It depends on lots of things. The ambient noise level, how loud is it? Are there ships going by? Is it windy? What are the propagation conditions? So can the sound go directly from the animal to another animal, or does it bounce around through the environment or a canyon to get from point A to B? And then thirdly, how loud is the animal calling? We all know this, you whisper if you don't want the person over there to hear. Right whales do that too, particularly on the calving grounds. And then you increase your volume to project further. And I will add to this that there's also the Lombard effect, which is a unique thing that mammals do. As we hear the ambient noise increasing, we automatically raise our voice to be heard at longer distances. If you've ever left a busy auditorium and your ears are ringing and your throat is sore, we can thank the Lombard for that. So what does this mean? Well, this means for conservation with right whales, we need to understand how far our systems can hear animals at all times. So this is, Dom's going to talk about this a little bit later, but while I was working at SMER Consulting, we have a real-time monitoring buoy for right whales that the idea is to place this around pile driving activity on the east coast of the United States to monitor right whales around offshore wind construction. So last year, about this time, we went out and we played back several thousand simulated right whale calls to understand the probability of detection for these instruments at this location. Dom is going to talk about that a little bit. No, he's shaking his head. Oh, yes, which way is he shaking his head? So he's going to talk about it a little bit more later, but that's something to keep in mind. And then the last bit that I want to talk to you about is some work that I've been doing off and on since about 2011. And this is to do with the Lombard effect. So back in 2009, Cornell Bioacoustics program put out 10 archival monitors, and some poor schlep annotated about 10,000 individual right whale calls and localized them all. This is a really cool data set because then we can evaluate the detection function in this area as a function with lots of different covariates, because for each of these localized calls, we know on which sensors it was and was not detected. So this is the take-home of that work. So I fit several models to our detection function data and found a complex relationship with the probability of detection here on the x-axis, on the y-axis, and the range on the x-axis, and some variables such as noise level. We would expect it to decrease linearly if it was no relationship, but the take home here is that we may be able to hear whales further than we think, and that can be good or bad depending on what you're doing. So we need to understand what our detection function is. If we overestimate it, we can put animals at risk. So if we think we can detect animals out to this far, but we can only detect animals this far, and we put our systems in a shipping lane, we may not be monitoring animals that are calling, and that can be bad. We can also bias our estimates on habitat distribution. So there's different ways of including that into our analysis that we can nerd out on later. That's about it. So I will take this time now to thank my co-authors on both these works, including Dom, who's in the field, and ask if you have any questions.
Thank you, Nikki. Dom, do you want to ask a question or give your talk now?
Dom very cheekily asked how far away you can hear a right whale call. In Cape Cod Bay, with noise levels in about 102 dB in the 20 to 200 hertz range, your 50% of your calls are going to be detected within 15 kilometers.
And I guess I'm just wondering the implications of your research for detecting killer whales, humpbacks within our shipping lanes?
So these were more methodology approaches. So we are at Smear Consulting looking at evaluating the caboose or the cab guardian systems with respect to killer whales as well as localization accuracy.
Great. Thanks so much. And our next speaker, and I should check, Linda, is it okay if you give your talk right after lunch? Are you first up? Or you want to get it in before lunch? Okay, we'll try before lunch. Carol Morales is our next speaker.
Thank you,
Carol.
Andrew. Good morning, everyone. My name is Carol Meirelles. I'm from Brazil, and I have been working with marine mammals for a while, especially manatees, and I'm really glad to be here today to talk about my research project here at the Marine Mammal Research Unit, UBC, under the supervision of Professor Andrew Trites. So a little bit about manatees. You probably don't know much about them. They are part of this order Sirenia. There are only 5 species in this order, and all were intensively hunted and are still hunted for subsistence. One of these species is considered extinct, and the other 4 are classified as vulnerable by the IUCN Red List. Here you can see a map with the Sirenian's distribution. In the right corner, the area that I am going to work with manatees. And an interesting fact is that you had a Sirenia species here in the North Pacific Ocean, the stellar sea cow that unfortunately is now considered extinct. Well, back to South America, there are 2 manatee species there. West Indian manatee that occurs mainly in coastal and estuarine waters, and the Amazonian manatee that occurs in lakes and rivers of the Amazon basin. Both species are still hunted and also face several other threats like incidental captures in fishing gears and habitat loss. So the main question of my research project is which are the priority areas for manatee conservation in South America? And how I'm going to answer this question. First, investigating both species distribution and suitable habitats using species distribution modeling. And for this, I need evidence of manatee presence, obtained through land-based surveys, boat surveys, strandings, interviews in coastal, riverine, and indigenous communities, telemetry, indirect evidence like feeding trails, and also aerial surveys. So this is how looking for manatees looks like. Looks pretty easy to get evidence of this manatee presence, right? Unfortunately, this is in Florida. Yeah. In South America, this is how looking for manatees looks like. Yeah. Especially in areas where the manatees are still hunted, they only show the tip of their nose to avoid to be hunted. So if you have lucky, you can have this. If you have a lot of lucky, you can have this. Yeah. And so because of this, a lot of researchers are using siding scan sonar to detect manatees in Brazil and also other South America countries, especially in rivers and estuaries. This is a project that I was part of. And here you can see on the right side the sonar image, and there are 3 manatees on the right side of the boat and 1 manatee on the left side of the boat. And you can be thinking, oh, come on, it can be that hard to find these big, slow, and cute marine mammals, right? And I will tell a story about how hard it can be. 20 years ago, I was an intern in this project in the Amazon forest. I lived 4 months in this floating house. And I was tracking manatees through radio telemetry, 4 manatees, Amazonian manatees. And I knew the manatees were there because I could hear them, okay, by the equipment. But I also wanted to see them. And I spent 4 months every single day trying to see them. 4 months. And this is what I got. Yeah, so it can't be that hard. So how to deal with this? Well, of course, manatees have been studied for decades in South America, right? And there are a lot of information published in papers. Some information you can find in online databases, theses, and also reports. Even so, data are still lacking on manatee presence because information like the geographic position of the observations cannot be found in these sources. And most of the data is still in investigators, researchers, and institutions' database that are not accessible. So to avoid to go to field and spend a lot of time and a lot of money looking for manatees, how we can work with this? Well, you're gonna work in collaboration. We invited 35 manatee knowledge holders from South America that have been studying manatees to provide this presence data to us so we can investigate the species, both species, and identify their critical habitats. So this project is just beginning. I don't have any results yet. We are receiving the data and identifying what kind of data we're going to use. And we're also going to meet with the knowledge holders to discuss and identify manatee habitats and which variables we are going to investigate in the models. So that's it. Thank you so much.
Thank you, Carol. Linda, want to come down? And oh, you're not on the agenda? Wait, I did the wrong one here. Who is up next? I did, unless you... I'm sorry. Miscommunications. No, maybe, Linda, if you're fine with it right after lunch, then let's do that, because I don't want to also keep people longer. What would you like? She's ready now. Okay, you're ready now, Linda. Thank you. And just while we're waiting, Carol, can you tell us a story about who is that manatee on the screen?
Oh, this is, it wasn't on the last slide. It was Alva. We have a huge problem with calf strandings in Brazil. There are a lot of calf strandings because habitat loss, and I coordinated a manatee rehabilitation center for 10 years there. And this was the first calf that we rescued there. And she's now back in the wild. Yeah.
Thank you.
And this would just be if I wanted to go back or something? Sounds easier.
Yeah.
Okay, I think it sounds easier. Okay.
And our last speaker before lunch, and lunch is here, is Linda Nickel.
Just a pressure cooker. I know. Hi, I'm going to talk quickly about sea otters. I'm going to give a bit of an update. Some of this I've presented before. Yeah, so getting right onto it. So we at Fisheries and Oceans and people before that have started doing surveys for sea otters on a fairly regular basis since 1977. And at DFO, we've been doing it consistently since 2001. And this is just to show you a quick artistic rendition of sea otter range expansion since 1997. This is where they were at that time. We'll just get this out of the way. So this is the range of sea otters based on where we observe rafts during our surveys, which we do in the spring and the summer. So this is up to 2017. And so by surveying, we go out and we count the population and we can document the range and the distribution of the species. And just ever so quickly, just why do we do these surveys? There's a bunch of reasons. Initially, when they were first introduced, you can think that the first surveys were to assess whether the colony had actually established, whether they persisted. It's really important and was really important to assess their conservation status as an endangered species at the time. When they were first introduced, they are now special concern. It was to begin measures to look at trends in population growth and range expansion, so that's a long-term data set that we now have.
And really importantly, it was also to document changes along a gradient of sea otter occupation time, which has been a foundation of many studies looking at ecosystem changes that come along concurrently with sea otter occupation. And as time has gone on, this has included other things like looking at ways to be able to provide abundance estimates as we go forward for questions around conservation or management decisions with this population. Still assessment for Kosiwik, keeping that time series going, which is the foundation of it all. Providing distribution, abundance, and trend data that supports questions around ecosystem and fisheries effects. And providing information for oil spill response. So where are sea otter rafts on the BC coast is really important for that type of work, and that needs to be maintained. So these are kind of a constellation of reasons why it's worth doing. So ever so quickly, a visual here of the survey effort. So in 1980, which is one of the earlier surveys, you can see a little squiggle of lines south of Brooks Peninsula. Is that showing my, yeah, which is where that's how much effort was required to survey the population. By 2017, this is the sort of vessel track effort that we need to be able to survey the population. So it's quite a lot of effort to get out there and survey the whole population. Back to what we've learned. So what we've learned is that the sea otter population has grown a lot, and that we know from BC and other areas that when they first occupy an area, population growth can be very rapid. It can be at the physiological maximum of the species, which can be as much as 20% or more a year. In the initial stages of population growth, when there's no density-dependent factors at play, sea otters are food limited. That's one of the main drivers of density dependence. And then after a while, we began to realize that our simple linear regression didn't fit anymore, and we moved to using a piecewise regression, because by about 1995, it was evident that it didn't really work. So it looked as if population had slowed overall. But in fact, what this was telling us was that there were density-dependent processes happening within the population. So somewhere in that range, there's a range of different things that are going on. Okay, it's not moving. What's going on here? There we go. Okay, and so what we know is that sea otter, well, populations grow, they grow, and there's two sort of simple models that can be envisaged to think about how populations grow, and these are exponential. So population grows without limits. It's density independent. And that's what we see in sea otter populations, as I said, when they're not food limited. So when they've come into an area that they haven't occupied for literally 100 years. And that over time, population slows. This is commonly what we find with species. They are limited by something. It could be nesting habitat, it could be spawning habitat, or it could be food. And so in the case of sea otters, the population slows as it approaches an equilibrium density with the habitat. So these are two models that we can think of to describe what's going on in the population in different parts of it. And the reason this is happening in the sea otter population, the reason it's not the same everywhere, is because sea otters have small lifetime home ranges, so they don't move very far in their lives. 20 to 25 kilometers is their typical range. This is based on studies from various places. I've thrown in a few references here. They have limited dispersal distances over the course of their life, even when they do disperse. They're feeding on sessile prey, so their prey doesn't move around. And what we can observe when we're looking at them feeding in areas is that their diet actually changes as the population increases. So diet changes with increasing population density and occupation time. As the population density increases in area, time spent foraging to acquire calories increases, and also energy intake rates decline. All these things are things you can track with population status in an area. And together, these diet things, which relate to food limitation, and then these habits of not moving, create population structure at small scales. So what happens is different areas are experiencing different demographic processes. And so we now look at sea otter populations instead of just looking at it overall. We've started to look at it in sub-regional context, where we look at growth rates in different parts of the range. And so you can think of that earlier graph where I showed the logistic growth curve and the exponential. And parts of the population, not surprisingly, are where they've recently expanded, are still growing exponentially, but they represent relatively small parts of the population. So the little pie charts show the proportion of the population in 2017 that was comprised of, say, subregion 10. And areas that have been long occupied are approaching these equilibrium densities, which fluctuates with the annual availability of food in the area. And the way this works is actually through pup survival. So pups are born, but the proportion of pups, or the likelihood that a pup is going to survive after it's weaned, is lower in areas where populations are near this equilibrium density. Okay, and here's another way of looking at it, which is there's this inverse relationship between years of occupation and the average growth rate. So the longer an area has been occupied, you have more otters, and growth rate slows down. And so just to let you know the way we do those sea otter surveys, I showed you that slow trail of tracks on the coast. We do most of the survey work by small boat. We have done some aerial work, but mostly it's done by small boat with observers following consistent routes through our survey segments. And I don't have any new data. We have been very busy over the last few years, though we do surveys every summer in portions of the range. This map is just to quickly show you, there's a lot of colors here. Essentially, I've got all the survey. We have 24 survey segments on the coast, and the colors are just alternating so you can see the different segments. But the main thing is that these darker ones are the ones that we've managed to survey last summer and the summer before. So we've managed to survey all of this. We also completed a survey in this area, which is new. It's an area, we're always interested in the edge of the range where the population may be expanding. So we've surveyed this area as well. And then these are areas of the coast that we have yet to do to get another complete survey of the coast, like we did in 2017, except this one is going to be done over multiple years. And that's all. That's it.
Thank you. You can hang on to your mic because you are our last speaker.
Oh, right. Okay.
Just one question. Do you expect sea otters to eventually expand the range into the Salish Sea?
You know, I think there's been thought that no, they didn't in the past. They probably won't. Perhaps paralytic shellfish poisoning, which would be more prevalent in the Strait of Georgia, has kept them out. But you know, I'm not sure. Mm-hmm. I'm not sure anymore. I think we'll just have to see.
Yeah, that's great. Okay, we are going to take a break here for lunch. We'll be getting back at probably 1:15 hours, 50 minutes later than scheduled. And Dom told it to be first stop. You don't want to miss it. Thank you.
get signal or not? Do we have signal? Do we have a signal? Yes? Okay. Check signal. Okay.
Welcome back, everybody. We'll get started once again. We've got an afternoon full of also interesting talks. And I'm going to start off first here with Dom Tollett. And I do want to point out to all the speakers, there is a timer up here on the right, and it counts you down. And so please keep an eye open for that as well. And I'm not sure if we're missing something.
My slide.
No, just stand by. And I think we're ready. Okay, the floor is yours. Although we're not up there yet. Oh, yes, we are.
I'm going to talk about acoustic monitoring studies in the Salish Sea. For those that don't know, SMRU Consulting is the commercial offshoot of University of St Andrews Sea Mammal Research Unit. I'm one of a team of 22. We do monitoring, modeling, and risk assessment studies for a variety of entities, and working on offshore wind, tidal, infrastructure projects, and noise effects. 13 papers in the last couple of years, 6 of which have been focused on Canadian species. Today I'm going to talk about a newly developed coastal acoustic buoy, CAB, which with funding from US Department of Energy and Maryland DNR, focused on offshore wind mitigation. This new tool that we call the CAB Guardian has 3 hydrophones, so that allows the ability to track or localize cetaceans as well as giving you noise levels. Comms is via Wi-Fi, 3G or satellite link, and the cabs have been pioneered by Jason Wood, who is in our San Juan office. And Caitlin gave you an overview of our first full system test with 5 cabs last year, and she kind of showed that getting bearings and localizations improved the efficiency of mitigation measures for offshore wind piling for right whales. And Caitlin's got a paper in MEE that I direct you to. I'm going to talk about some of the studies in the Salish Sea that we've used this cab. First one was with Frankie Robertson in San Juan County, where we were monitoring off South Beach and showed that presence of boats or the presence of echo sounders increased noise levels by tenfold, a 10 BD difference. We also showed high usage of South Beach, similar to Lime Kiln further north, as well as actually an increase in noise due to COVID because boats, US boats, were actually stopping at the border and hanging around San Juan rather than coming into Canada. In each of these slides, the location is marked in blue. The red dots are some of our acoustic monitoring locations. The next study is ongoing at the moment and just working to monitor a slowdown that Quiet Sound is undertaking in Puget Sound for the last couple of months, or it'll be for 2 months starting in October. And there's been a number of superpod sightings down in Puget Sound during this slowdown. So again, trying to reduce noise, and the cab's being used to look at that reduction in sound, as well as tracking vessel speeds and killer whale detections. We've been working with DFO, specifically Harold Yerk, to assess winter usage by killer whales in the Strait of Georgia with a node near Point Roberts. And we're also testing, or Harold's testing, concurrent use of thermal image and PAM detections at Kamana Point, which is part of his strike risk reduction program. And lastly, we've been putting cab brains into what are called uncrewed surface vehicles, and that's a company out of Victoria called Open Ocean Robotics. So you can now have mobile passive acoustic monitoring. I'm now just going to briefly go through work that we've been doing with the ECHO program, Vancouver Port Authorities, enhancing cetacean habitat group. And the first thing I'm going to talk about is a kind of surprising detection of northern residents up in Indian Arm. This occurred 7th of February in 2022. It was the A42s from A-Clan, and I guess they were heading up there for Chinook or chum, but this was certainly surprised us to find northern residents so far up into Indian Arm. Although I should note we have been detecting southern residents in English Bay as well as transients up Indian Arm as well. We did a similar analysis to Chloe, looking at night and day differences in killer whales and porpoises at Lime Kiln, and hopefully you can see here that both transients and porpoise show a strong nighttime activity or presence, whereas southern residents, the graph on the right there, are showing a more daytime presence at Lime Kiln, and those are all statistically different. And lastly, we're putting in an array at Lime Kiln, so we'll be able to track whales' direction of travel next month. As I mentioned, the ECHO program has been running vessel slowdowns through Harris Strait for the last 5 years. Last year, we monitored the reduction in noise and assessed that there was a 3.7 dB reduction due to the slowdown, which is a reduction in noise intensity of 57%. And that's occurring both at low frequencies and at higher frequencies. And I'm happy to say we were also doing real-time detections for killer whales during that Aleutian Isle spill, given the Coast Guard up-to-date knowledge of killer whale presence. It's been a banner year for the ECHO program in 2022. They've slowed down or moved more than 5,700 vessels this year over a 5-month summer slowdown period. and we monitor both the noise levels and the speed through water using AAS and a UBC tidal model. This year we're also looking at co-benefits. What's the benefit to strike risk reduction and emissions due to vessels slowing down in these areas? And lastly, I'm going to end with an expose where we're raising a red flag over an increasing use of ultrasonic anti-fouling devices. These devices are around 23 kilohertz, and hopefully you can see those bright red lines and purple lines showing these high source levels of these devices. They're clearly in the hearing frequency, peak hearing frequency of killer whales, with clear disturbance and masking potential. At the moment, its use is unregulated. The industry is aware, but obviously it is a dilemma because paint options also have biological concerns. And with that, I'm just a minute over, and thank you very much.
Thank you, Dom. Questions, Paul?
Hey, Dom, what's the source intensity of that ultrasonic?
Oh, yeah, please. Stand by.
The measurement we made was 187 dB from the vessel with the ultrasonic anti-fouling device on. There's a paper that's coming out by Martin et al. In Aquatic Noise that will give you some of those sound exposure levels. It's clearly higher than most echo sounders.
And Dom, compared to fish finders, is it directional? Is it omnidirectional?
It's wider, so those are often straight down, and this one is clearly lateral as well.
Thanks so much. And the speaker is Grace Bear.
Hi, everyone. My name is Grace Bear, and I work with the North Coast Cetacean Society, or better known, I guess, as...
Sorry, we just need to get your first slide up.
As BC
Rob?
Whales. No worries. Like I said, I work with the North Coast Cetacean Society, which is maybe better known as BC Whales. As an organization, we have been conducting whale research along the BC coast for more than 20 years, following the lead of our CEO and founder, Janie Ray. Before I get into any of this, I just want to quickly mention that none of our work would be possible, and especially the work that I'm presenting today, without the support and permission of the Gitga'at First Nations community in Hartley Bay. Throughout our field season, we are stationed right here at the Finn Island Research Station, though many of the work that I'll be presenting also extends into further reaches of the province. Our three primary study species are humpback whales, orca, and fin whales, so we do place a particular emphasis on data collection for those humpbacks and fins. In the interest of time today, I'll just be giving a very quick overview of some of our bioacoustics projects, as well as some aerial monitoring that we are conducting that, of course, incorporates drones. If you're curious about any of our other work or any of this in more detail, please just come find me. Beginning with bioacoustics, we are leading a multi-organization acoustic monitoring project all the way up and down the BC coast, and this is coined the Pacific Hydrophone Network. This is a phenomenal effort between both NGOs and First Nations communities, including ourselves, as well as the Gitga'at First Nation, the Kitasoo-Hey-Hey First Nation, the Heltsick First Nation, as well as Orca Lab and the Saturna Island Marine Research and Education Society. All the blue triangles that you're seeing here on these maps represent smaller networks of hydrophones that exist in those locations. This is a project that is still in its growing stages, and it's very exciting. The ultimate goal of this project will be to monitor whale acoustic habitat usage all the way up and down the coast, as well as tracking noise levels in these locations. Within our particular research area surrounding Finn Island, and within especially the region of Squally Channel up in northern BC, we are also partnering with WWF Canada and the Gitga'at First Nation on what's called the SWAG project. This is a three-organization collaboration, of course, and is aimed to reduce the threats to vessel traffic to whales within Gitga'at territory, as well as to increase the capacity for monitoring the impacts of this vessel traffic. One way this is being done is through the creation of a computer software that is able to accurately detect, classify, and localize the vocalizations of whales that it's receiving in real time. So as a lot of us know already, a lot of the sounds that are coming in that are produced by these animals are very unique, often down to a species level and occasionally beyond that down to a specific behavior or a specific call type. The computer is able to receive this information and then classify those appropriately. What this ultimately allows us to do is in areas like Squally Channel, where we have 4 hydrophones, is to go one step further than classifying and actually take into account the time delay it takes for each call to reach each hydrophone and to plot it on a map. So the vocalizations that you're seeing on this map are occurring over the course of 1 day in 2019 and is giving us a really good representation of acoustic habitat usage over the course of our field seasons. So similarly to our ability to identify the calls of whales, we are also able to identify the individuals based on unique pigmentation patterns and scarring that occur on their bodies. What investigating scars in particular is now allowing us to do is to investigate the pervasiveness of threats like entanglement and ship strike. So we are tackling this investigation by incorporating and comparing both DSLR photography as well as aerial imagery captured from drones. So the imagery captured through both of these methods is lending insight into the frequency of the occurrence of these events by looking at live individuals, as well as lending inference into where these events are occurring on the bodies of these whales. This method is also just generally encouraging us to get drones into the air, which is proving incredibly helpful in this case of active entanglement that's on the screen here, because this was actually not visible from a lateral perspective from the boat or from any of the photos that we captured with our cameras. Drones are also allowing us to accurately measure the body sizes of whales with the use of LiDAR technology. This is now also being used as a part of a ship strike risk assessment model that is being built and written for publication at the moment. The LiDAR data we're collecting will additionally allow us to track changes to the body conditions of individual whales that we encounter through time. Finally, we are incorporating drones into genetic data collection in collecting blow samples and sequencing the DNA that is trapped therein. This project is being led by our current PhD student, Adino Mani, who is currently in the lab in Copenhagen, sequencing the samples that we collected this past season. We're hoping that this genetic insight will lend us more information about the relatedness among the individual whales that are routinely returning to the productive waters of the BC coast, as well as if there's any genetic drivers that are lending us insight into sociality as well as foraging associations. So thank you for listening. I'm happy to take a few questions now or some more later as well.
Do you have any questions? Question up at the back. And Grace.
How do you find that adding extra weight to the drone decreases the battery life of the drone? Is it up there long enough to get the samples?
Yes, so the question was, does adding things to the drones like the Petri dishes to collect DNA samples, does that decrease the battery life? The answer is yes. Thankfully, what we're able to do is track the whale for a few breaths before it comes up to the surface to get the drone ready, so that once the drone's up, it's able to locate the whale it's going after and usually collect a few breaths of sample before returning home. So the drones we fly typically have about a 20-minute battery life.
Anything else? Okay, thank you very much, Grace. There we go. And our next speaker is Thomas Daniel Belcroze.
Yeah, hi. I'm with the Cetacean Research Program and with DFO, and I figured that since we haven't been here in a while, I would try to give you an overview of our various projects all along our shores. And I decided to go at this in a geographical fashion. So I've on this map, I've just plotted a subset of our effort track for just 2022. Thought it'd be good for you to see your tax dollars at work. And as you can see, there's a wide range of projects in there. I'm only going to touch on a few of them, and I'm going to leave others on the side because Linda already told us everything there is to know about otters. But you can see there's some wide-ranging small boat surveys in there. There's some systematic surveys at various scales, some inshore, some on the shelf, some offshore.
It's okay, we know.
All right. Not worried. And I'll start with the killer whales, but then I promised Anna that we're going to move to other cetaceans, not just medium-sized superstars or something. And so this is just a subset of our tracks that we use to keep a census of the northern residents. It's by far our oldest time series of data. And it's something we've made a few changes to in recent years. For instance, we've changed the way the information is disseminated. We felt it was useful to have it published in a yearly report that's on the CSAS website, so the Canadian Science Advisory Secretariat website, so that every year people can find it, find the right numbers, cite it, and see, especially since sometimes the information can change retroactively with the years. This is the result. So these tracks are from 2022, but it takes us a few months to process the data. So this is the result for the census in 2021. As you can see, the plot on the right shows you the population has continued to increase, plus 2.5% since last year, which is pretty much in line with the trend since the early 2000s. Our best estimate of this population at this point is 332, but in 2021, there is 22% of the groups that we could not find or they were not encountered, and so that creates uncertainty. And that's really a reflection of something that's been going on that's very similar to what Linda was saying earlier about sea otters. As the range and numbers of this population have continued to grow, it creates a logistical challenge to find them all and census them all in a given year. The plot on the bottom left is an excerpt from a talk we gave at the SMM conference this summer. And basically it's just showing that if you only looked at animals, so the groups of animals that we find in the historical core area highlighted in orange on the map, back in the 80s, you would see 100% of the groups in that area. But nowadays, if we only did that, it would be about 50%. And so we've become very reliant on effort in other areas like Hattagwai, the central and north coast, and also the west coast of Vancouver Island. Another type of work we do is we call them large whale surveys. And we try to go offshore with the Coast Guard ship Tully every year. And we do this for a variety of reasons. One shift we've made in the recent years is to move to a more systematic pattern of doing surveys along systematic transect or zigzag lines. And doing this allows us to provide more quantitative evidence to answer some of the stock assessment questions that are coming up. So on the right, for instance, you see a publication from last year by Brianna Wright showing density surfaces that from our 2018 coastwide survey. On the top there, you see a report that was just a paper that just came out 2 weeks ago. It's a collaboration with JASCO and Parks Canada, and it compares some of the acoustic moorings we've been deploying with some of the sightings we've been seeing on these visual surveys to better understand the distribution, the year-round distribution of cetaceans. But one of the fun things I wanted to also briefly mention is some of the things we've seen on offshore. So those green lines on the bottom left, they're from our 2002 offshore survey. And again, this year, we saw a lot of sei whales. And we saw a lot more even the year before. But prior to that, we would almost never see them, even when we were out there in good conditions. So we've been surprisingly learning a lot about density, population, and habitat of sei whales in recent years. And are trying to see if we can understand the pattern that explains why they're there some years and not others, something that the whalers seem to have noticed a long time ago. Something that's new too, and I thank some of our gray whale talkers, speakers from this morning for introducing it, is that the status of gray whale might be changing in Canadian legislation. It was Kate this morning who mentioned that the Acoustic has recommended that the Eastern North Pacific population be split basically into two different designable units, and one of them, the Pacific Coast Feeding Group, should be considered endangered. If species at risk basically accept this recommendation, and there are public consultations going on right now, then it will mean that we'll have a mandate to identify critical habitat and monitor this population going forward, which is why we try to be a step ahead of the curve. And with Erin Foster and Linda, we've tried to start some gray whale work on the west coast of the island with the help of Jim Darling, Wendy Stanislow, and Brian Gisborne, trying to collect photo IDs, biopsies, prey, and fecal samples to better understand how the prey affect which habitats are important for gray whales. And finally, Something that has also been going on for just the last couple of years, in the context of the recommendation 5 and 6 for the TMX project area, we've been asked to monitor more accurately the area between Vancouver and Swiftsure. And so we've tasked Christy McMillan with a project to do systematic transect in the entire area every month for the last 2 years. And the idea behind this is that there's a lot of work going on already in this area, but not enough of the systematic type of work that we need to answer some of the questions quantitatively. So as you can see, this is just an example of humpback whale distribution in, that would be something, humpback whale distribution in the Salish Sea, and you see those monthly plots, and then turning this into quantitative estimates on the bottom right, you can see harbor porpoises, Dall's porpoises, and humpback whales across seasons. And we hope to continue this. I mean, preliminary data is also already available as a technical report, but we hope to continue this further, combine it with satellite tagging, or not satellite tagging, suction cup tagging, and acoustics to get a better sense of the vulnerability of these animals to ship strikes in the area. Thank you.
Thank you, Toma. Any questions? And I guess, Toma, I was curious if you're seeing sperm whales. I wasn't noticing them in your offshore surveys.
Yes, we do. They're few and far between, and some years we seem to have the right conditions to see sperm whales and beaked whales, and some years we don't. I suspect that if we really wanted to see more, we'd have to focus on some areas of particular characteristics, like canyons and other areas. We were hoping to hear more when we did the 2018 coastwide survey. We had a towed array, and we were hoping that we would find more, but we didn't find as many as I thought we would. So I think acoustics would be better methods for them anyway.
And one other one, just in terms of sei whales, is there a theory why you're seeing more sei whales now than before?
Well, obviously effort is a big part, because we didn't used to go this far this often than we have in the last few years. But still, there are years, like 2019, where we were out there in exactly the same place with perfect conditions, so beaked whales, but no sei whales. And then a couple of years later, we see plenty. No, I would imagine it's incursion of their favorite foods, but what that is exactly, I don't know.
Okay. Thanks very much, Toma. Sorry, we're just going to take 1 second. It looks like we're trying to get something set up here and running.
We've, unfortunately to everyone online, sorry for losing the video. Our video capture card has decided to stop working, so we may need to reboot that while we're taking this presentation, unless we want to take a break. That's up to Andrew here.
Are we minutes away, or can we do it on the fly? No idea. Just give us 2 seconds here. We're consulting with the Wizard of Oz. Just stand by. Aaron has extra time.
I was going to say, I'm still going to try to go as fast as we possibly can.
That's for you, not for me. This happened to us twice when we were setting up, and it's been okay so far. All I had to do was touch that cable, and now it's dead.
It's me, Rob. It's okay.
All of you? Okay. As long as we're clear on that.
I'll take the heat.
Just give like 2 minutes here, and we'll see if he settles down, because you'll also be distracted as you're talking. He's like pulling cables.
Well, waiting till late is nerve-wracking, but a lot of folks have already talked about some of the things we talked about, which is good. It means we can, skip some time here.
But I think you're also at ease with the crowd, so I'd be more worried if you were like first year's master student.
Well, that's very flattering.
Standing up to...
I feel like one inside.
No, we lost the feed onto the screen. Oops, we're back up.
We're not good. We're not in a hurry. Sorry.
We just haven't abandoned you and gone to sit down.